Okay, so this is the lesson we're going to do, osmoregulation and excretion. And that is, just like I told you before, a part of the 5th unit. So let's move on. Now, before we start the lesson itself, there are some learning outcomes that the government or the Department of Examinations want you to know. They have given this. I hope you have gone through the teacher's guide at least once, because in the teacher's guide, they have given the learning outcomes. You can always download it from NIE website. If you know what they're expecting from you, you can always prepare yourself to give good answers. All right. So these are the learning outcomes that they're asking from you. They want you to know, state what is osmoregulation, and you have to define osmoregulation, excretion, then relate the importance and need of osmoregulation and excretion for survival of animals. Describe relationship between metabolic substrates and excretory products. Name the nitrogenous excretory products. Describe the relationship between nitrogenous excretory products and the living environment. And state examples of organisms which secrete different types of nitrogenous excretory products. So in the first part of the lesson, these are the learning outcomes, or rather these are what they are expecting you to know in the first part of the lesson. Okay, so the first question in your tut is, what is osmoregulation? Children, in biology, it's not like in physics. You don't have to think out of the box a lot. You have to know the facts that have been proved scientifically. And so, especially when it comes to the definitions, you have to be a bit more accurate. You can't write what you think, you can't write in your own words. They have given definitions, and it would be much accurate if you can write the definition that is in your resource book. So if they ask you what is osmoregulation, the answer would be, osmoregulation is the process by which organisms control solute concentrations and water balance within the body. So now, what is osmoregulation? If they ask you the definition like this, most of you, I have heard from students, what is so hard about doing biology is you have to remember a lot of things. There are so many definitions that you have to remember. That is true. But if you have a way of remembering the definitions, now I myself, when I studied, I always broke the definition into questions. Let's take this for example. If they ask me, what is osmoregulation? I will break the answer. What is it? It is a process. Then who does it? Organisms. What happens? Controls solute concentrations and water balance. Where? Within the body. So, what is it? Who does it? What happens? And where it happens. Those are the 4 parts in this definition. Osmoregulation, it is a process by which organisms control solute concentrations and water balance within the body. I'm pretty sure if you study the definitions while breaking them down into questions like this, it would be much more easier for you all to remember the definitions. So osmoregulation, children, it is as in osmosis or osmotic pressure. And this is about maintaining a constant osmotic pressure inside organisms. Why we have used the word organisms is because it is not only about animals. There are microorganisms, plants, and animals that do osmoregulation. So if you write the osmoregulation is the process by which animals control, then your answer is incomplete. Okay, so remember that it has to be organisms. So things like these, children, while you're studying, you can ask yourself, why have they given organisms instead of animals? And you can prove the reason as to why, so that you will remember the definitions much better. And children, in our body, especially in the brain, there are osmoreceptors in the hypothalamus in the nervous system lesson. That is the other next part of the animal form and function lesson. You will learn about these uh, osmoreceptors and about the hypothalamus. So in the hypothalamus of our brain, there are osmoreceptors that detect the osmolarity or the solute concentrations of the blood that is passing through the brain. Those are like checkpoints in the brain. And when the blood is flowing through along these osmoreceptors, if the solute concentration is more or greater than normal levels, then they will detect it and they will inform the hypothalamus. So in humans, why we have osmoregulation is because we have to ensure that our total blood volume and the dissolved substances in the plasma and tissue fluid remain constant within a favorable range. Simply, osmoregulation is a balance between the solute concentrations in the body and the water levels. All and everything done to maintain homeostasis. Homeostasis, that is the condition where even if there is a drastic change in the external environment, our inside body will not change and will maintain relatively constant. Okay, that is osmoregulation. So I hope you all are writing down if you have printed, or otherwise if you have a soft copy, please do write down the definitions so that you can print it later after the seminar and write down, because I want you all to have a complete and you can study the entire lesson from this script. All right, so that is osmoregulation. Then the second question they have asked here, name 2 simple unicellular organisms that use contractile vacuoles for osmoregulation. You can see in the presentation, simple unicellular animals such as amoeba, paramecium use contractile vacuoles for osmoregulation. Who are unicellular animals, children? Unicellular, uni means one. So those who have one cell, simple microorganisms like amoeba and paramecium. For them, they don't have complex systems for osmoregulation. They just have simple contractile vacuoles. So as you can see, in this picture, this is amoeba with pseudopodia or false feet, and it has a contractile vacuole, and paramecium, which has cilia all around it, also has got contractile vacuoles and foot vacuoles. So for osmoregulation, even these small unicellular organisms have some way of balancing or controlling their solute concentrations and water balance. And why? Why do they have contractile vacuoles? You can say they're very simple organisms. Why do they have to have contractile vacuoles for osmoregulation? That is because, children, usually amoeba, paramecium, these kind of microorganisms, they are found in fresh water. And when they're in fresh water, sometimes the solute concentration of these organisms may be more than their environment or the fresh water. So what will happen if the contractile vacuoles are absent? Amoeba will fill with excess water. You know, there is something called a concentration gradient. We'll be learning that in future slides. So the concentration gradient is when there is a higher concentration and a lower concentration, and usually when it's along the concentration gradient from higher to lower, the substances will flow. So water too, if water levels are higher in fresh water than in amoeba's body, water will flow from the fresh water into the body of amoeba and it will swell up. If it doesn't have contractile vacuoles to remove the excess water, it will swell up and burst. Right? So in order to prevent bursting, amoeba itself has contractile vacuoles in it. So different animals have developed different structures for osmoregulation. The chemical reactions in the organisms result in the formation of waste products, which are toxic, children, and must be removed in some way. So then, what is excretion? This is the third question. What is excretion? And the answer is the removal of nitrogenous metabolite and other metabolic waste products from the body. So, why have they said nitrogenous metabolite and other waste products? In the definition itself, there are two parts: removal of nitrogenous metabolite and other waste products. The reason is, children, in our body, when there is excess nitrogen, those products like ammonia, urea, and uric acid, they are removed as nitrogenous excretory products because they have excess nitrogen. But urine is not the only thing that is excreted from our body. From the lungs, carbon dioxide is excreted, and from the skin, sweat is excreted. So other metabolic waste products are carbon dioxide and sweat. Therefore, if they ask you excretion, you can't only think of your kidney. You have to think of your lungs and your skin as well. Therefore, when they ask you what is excretion, remember it's a removal of nitrogenous metabolite and other metabolic waste products from the body. All right, children, I'm reminding once again, if you have any questions or if I'm too fast, you can always put your questions in the chat. Then why is defecation not considered under excretion? You might have wondered why, if defecation or passing down of feces, why is it not a form of excretion? Children, in defecation, that is the removal of undigested food. And undigested food does not go under any sort of metabolism within the body. Therefore, if something is not gone through metabolism within the body, we cannot take it under excretion. Therefore, remember, removing of undigested food from the gut is not a form of excretion. Always remember. And in many animals, excretory and osmoregulatory systems are linked structurally and functionally, especially the shark. You will remember as we go in the future slides that shark has a very nice system in which its osmoregulatory and excretory systems are linked structurally and functionally. Then, briefly describe the importance of osmoregulation and excretion. Importance of osmoregulation and excretion. Children, animals do not have a cell wall, right? You have learned even in your O-levels, and might be even before that, that animals, they don't have a cell wall unlike plants. So they only have a cell membrane as their outer covering, and hence don't even have a good shape. They just have the shape that their cell membrane is giving them. So when they don't have cell wall, they have to have a way for effective body functioning and survival. To maintain a constant internal environment, children, there has to be a favorable balance between concentrations of water and the solutes. Okay, So if I'm just going to describe you what an internal environment is, it refers to the fluid which allows life-sustaining exchanges and surrounds the cells. Internal environment is the surrounding that covers our cells, children, that helps in exchanges between the blood and interstitial fluid. Okay, so in order to maintain this internal environment constant, we have to we must use osmoregulation and excretion. Okay, so we can say, if you now we have asked you to briefly describe the importance of osmoregulation and excretion. You can say for effective body functioning and survival, you can tell the first point, animals have to maintain a constant internal environment, especially relative concentrations of water and solutes within favorable limits. And then you can say, therefore, animals have to regulate the chemical composition of body fluids by balancing uptake and loss of water and solutes to prevent the end products of excretion being toxic. That is, when, like I told you, nitrogenous end products contain excess nitrogen that is not good for the body. And these toxic products must be removed from the body as soon as possible. Otherwise, they can harm the body and even lead to denaturation of proteins. In order to prevent the harming of the internal environment of our body, osmoregulation and excretion plays a major role. Okay, and you have to say that animals do not have a cell wall. Therefore, for animals, osmoregulation and excretion is very important. And just like I told you before, I told you that we'll be talking about concentration gradient. Yes, the driving force of loss of water in animals, as in plants, is the concentration gradient of solutes across cell membrane. Right? A concentration gradient is when the concentration of particles in one area is higher than the other. So the driving force for the loss of water in animals, just like in plants, is this concentration gradient. If a cell bursts, children, I don't think they can, because amoeba and paramecium, they're very simple structure, just unicellular organisms, children, just one cell. And if they burst, it's going to harm them. I don't think they can regenerate, because there are some organisms that can regenerate, and you'll be learning in the reproduction lesson. But ones like amoeba and paramecium, I don't think so, because the salinity or the solute concentration would be too high in their bodies. And if they didn't have these contractile vacuoles, it would fill up with a lot of water in order to minimize the solute concentration along the concentration gradient, and it would harm them a lot. And so animals have evolved different osmoregulatory strategies depending on the environment in which they live for their survival. Okay, so all of us, the main purpose of us is to survive. And I'm pretty sure you know Darwin, who has said that the ones who can survive, the ones who have the intellect to survive, will be selected naturally. I'm pretty sure you have learned all these things in evolution lesson, right? So the nature itself, the environment itself will help them survive by natural selection. So all of these strategies are evolving over generations. You have learned about evolution, change of genetic composition, and these exactly survival of the fittest. And so for the survival of the fittest, these osmoregulatory strategies are evolving over generations, right? So and then the next part is in the you can look in the slide. Why should animals get rid of toxic products produced in metabolism? Once again, in order to safeguard the composition of their internal environment. If there's anything inside our body that is harmful, we have to get rid of it. Simply, all of these excretory products, excretory end products will become harmful and toxic. So, main reason is we can't live with things that are toxic to our body. We will remove them. Okay. Then uh, again, now if we further explain children, all of these are given in your textbook in a long paragraph. So I thought I'll break them into slides and explain to you. This is one example. You know, when proteins and nucleic acids are broken down within the body, within the cell, during metabolism, the amine group in proteins, you have learned about proteins and nucleic acids. The amine group is converted to ammonia. Ammonia is a weak base and might be highly toxic if it is not within the levels. Anything that is above the levels will be highly toxic, children. So ammonia will be a weak base. Glucose oxidation during metabolism will release carbon dioxide, which is a weak acid. Accumulation of these weak bases and weak acids will harm or will alter the internal uh, environment balance. Okay, so this is the reason why we should, remove the toxic products from the body, and why osmoregulation and excretion is important. Like I told you, changes in acid-base balance will lead to denaturation of proteins. So in order to maintain the internal environment within their favorable limits, we have to always do osmoregulation and excretion. When we intake as food, it's not toxic. It becomes toxic, yes, because when we take food, children, we know things like fast food and things. Now, let's say you take food and uh, they might contain harmful substances like MSG, or there are a lot of things that are harmful to our body, and these things get metabolized in the liver, right? And liver is involved in detoxification. So, and the drugs that we take, not only food, drugs, and there can be a lot of things that can be harmful to our body. When we take these and these get broken down, I'm talking about metabolism, children. At the end, you have learned in digestion that every carbohydrate will break into glucose and the monosaccharides, every protein into its amino acids. So when they are broken down, carbon dioxide, let's say the oxidation of glucose results in carbon dioxide, and protein metabolism results in ammonia, just like what I told before. Now, if there's a lot of these things greater than the level they should be, then what will happen is they will become toxic. Just when you take food, maybe it won't be toxic, but inside your body, there are a lot of chemical reactions taking place, and sometimes they will be toxic. So all of these toxic substances, that is extra, whatever is extra more than the levels, will be released from the body. Remember that even your proteins might get denatured if your acid-base balance. Food poisoning, children, food poisoning you will learn in the microbiology lesson. There are also microorganisms. We can't forget them, right? In your food, there can be microorganisms, and these microorganisms itself, some of the microorganisms will already spoil your food, and some of them will go into your body with the food, and then they will cause different chemical reactions causing food poisoning, like Salmonella typhi. There are so many examples. You will get dysentery, cholera, like so many diseases are there. And food poisoning is a broader concept, children. It can be caused by microorganisms as well. So here, what I'm talking is during metabolism. We'll be learning about metabolism in the future slides. During metabolism of carbohydrates, proteins, nucleic acids, and fats, They will break into their smaller components like H plus, ammonia, carbon dioxide, K plus, and some of these may not be necessary because they might be greater than the levels that they should be. So those kind of end products should be excreted from the body, and that is why osmoregulation and excretion is important. That is why we have developed different structures for osmoregulation and excretion. Okay, so I'm pretty sure you all know the pH. We always have to have a favorable pH in the blood. It's around 7.4 in the body, the blood pH. So if something is extra, the pH will change. So something has to be done to remove whatever that is toxic in the body. Okay, so jot down all the points as much as you can, children, and then make a nice note to the question. The next question, what are the metabolic substrates in the cells? So the metabolic substrates in the cells, there are 4 of them: carbohydrates, fats, proteins, and nucleic acids. You can write them down: carbohydrates, fats, proteins, nucleic acids. These are the biological substances as well. So these are the metabolic substrates. Okay, there's a question, what will happen if pH is changed? It will balance normal, dear. That is because in blood there are buffers, things you call buffers. They will help in balancing the pH. But because our body is very sensitive and this homeostasis has to be maintained, somehow our body will react in some way to bring this back to normal. That is why we have this osmoregulatory organs to remove them as quick as possible if they're toxic. It will balance to normal the pH because of buffers. All right. So what are the factors on which the excretory products of these substances vary? There are 4 factors on which the excretory products of these carbohydrates, proteins, lipids, and fats will vary according to the chemical structure and the composition of our metabolic substrates, according to the availability of enzymes in each and every organism, according to the oxygen availability in the environment, and the habitat in which the organism lives. According to these factors, children, the excretory products of the substrates will change. We will learn this in the next slide. Don't worry if you don't understand. Write these down. Okay. Osmoregulatory molecules, the buffers. What do you mean by osmoregulatory molecules? Now, ammonia and carbon dioxide, if you take, they are, if that's what you're meaning, yes, sometimes now when we are moving on to urine formation, we will see that how buffers will act and how the levels will be balanced, how urine will be concentrated and such. We will talk about them later. And the time the meeting is ending, I'm hopefully planning to end this around by 4:30, and let's see how it goes, children. Just it would be between 4:30 to 5. Okay. Right. I hope you have written down. Now I'll explain each of these one by one. Briefly describe how carbohydrates, fats, proteins, and amino acids are metabolized. When carbohydrates are metabolized within the body cells, when oxygen is available, final excretory products are carbon dioxide and water. You have done photosynthesis, you have done respiration, you know how glucose is formed and how glucose is metabolized when there is oxygen. You have learned the huge process, right? And so You know the number of ATPs produced in when there is oxygen and when there is not, when oxygen is not available. So during aerobic respiration, when oxygen is available, children, the final excretory products will be carbon dioxide and water. And during anaerobic respiration, when oxygen is not available, there will be two forms of metabolism. Either carbohydrates would be metabolized in lactic acid, that the process will be lactic acid formation, or otherwise alcohol will be formed. So here in animals, generally lactic acid is produced. So when carbohydrates are metabolized, you have to remember only two ways. If oxygen is available, carbon dioxide and water will be produced. And in anaerobic respiration in animals, lactic acid will be produced, and the amount of ATP produced will be very less comparatively. Okay, so I told you 4 factors on which the excretory products of these substrates will vary. You can see one factor here, oxygen availability. When oxygen is available, it will be carbon dioxide and water, and if it's not there, it will be lactic acid. Okay, so then you will see how the availability of enzymes and the habitat of the organism also involves in the metabolism of these substrates. Fats, proteins, amino acid metabolism. Children, when fats are subjected to aerobic metabolism, the final excretory products, just like carbohydrates, will be carbon dioxide and water. Okay, because in when it's about fats, they don't have anaerobic anaerobic respiration. They enter the pathway at the citric acid cycle and cannot be broken down in the absence of oxygen. This means that if cells are not performing aerobic cellular respiration, the body cannot burn fat for energy. Okay, so for fats, unlike carbohydrates, fats can't be metabolized in both aerobic and anaerobic ways because they enter the pathway at the citric acid cycle. So for them, they only have aerobic respiration and produce carbon dioxide and water. For proteins, they contain amine groups in their structure, and during metabolism of excess amino acids, ammonia will be produced. And since nucleic acids also have nitrogenous bases, ammonia will be produced. Remember the structure of nucleic acids, they have a nitrogenous base. So ammonia will be produced during their metabolism. And depending on the habitat and availability of enzymes, ammonia will be further converted to other nitrogenous waste products like urea and uric acid. Okay, so now as a summary, for carbohydrates, if it's aerobic, it's carbon dioxide and water. If it's anaerobic, it's lactic acid and ATP. If it's fats, fats can only do in the availability of oxygen, so fats will turn into carbon dioxide and water. Proteins have an amine group. Proteins will produce ammonia in metabolism. Amino acids also, they have a nitrogenous base group, so they will also produce ammonia. And depending on the habitat of the organism and the availability of enzymes to convert ammonia into other products, ammonia will be converted to either urea or uric acid. So children, do you know what the excretory product of us humans? Is it ammonia, urea, or uric acid? You can put your answers in the chat. Okay, I would much appreciate if you can put your answers if it's ammonia or urea or uric acid. Very good, it's urea. I'm glad a majority of you know. For us humans, yes, it is urea. Okay. Then the next question, what are the 3 nitrogenous excretory products? So you know, ammonia, urea, and uric acid. Briefly describe the relationship between the nitrogenous excretory products and environment of organisms using the table. So So they have given a table. Before going through the table, before answering the question, we will simply discuss who excretes what. Okay. All right. So the next slide, ammonia. We'll first talk about ammonia. You can see images of organisms. I'm sure you all know who these organisms are. Since ammonia is highly toxic, a large volume of water is needed to excrete ammonia. Right? Ammonia, remember, out of ammonia, urea, and uric acid, ammonia is the highly toxic one. So a lot of water, large volume of water is needed if you want to excrete ammonia. Therefore, typically organisms that live in water, like bony fishes, many aquatic invertebrates, and aquatic amphibians, especially tadpoles, excrete ammonia since they have ready access to water. So all of these organisms who have access to water, who live in water, can excrete ammonia. So most of the aquatic organisms, like bony fishes, and I'm sure you all know who bony fishes are, right? Class Osteichthyes, those who have a homocircle caudal fin and have an operculum. All right, so bony fishes and aquatic invertebrates. Here I have given a picture of aquatic invertebrates and aquatic amphibians, especially tadpoles, will excrete ammonia as they have a lot of water alongside them. So not all the aquatic organisms excrete ammonia because the metabolism of different animals of animals is different. Some of the class of chondrichthyes, animals who live in marine environments, they will have a different excretory nitrogenous excretory product, right? And I told you, not only the habitat is involved here, the habitat, the availability of enzymes, right, also are involved when we are talking about whether it's ammonia, urea, or uric acid. Okay, so bony fishes, marine, many invertebrates, aquatic invertebrates, and amphibians like tadpoles will excrete ammonia. And the energy cost for producing ammonia is comparatively low. Why? Because ammonia is the one that is produced first during metabolism of protein and amino acids. Therefore, the energy cost for producing it is comparatively with urea and uric acid, it is low. Moving on to urea. Now, most of the terrestrial animals, they don't have sufficient access to water. So they can't have ammonia as their main excretory product. Therefore, most of them, what will they do? They will convert ammonia into urea and then they will excrete urea. So most of the mammals and adult amphibians will excrete urea. So I asked you a question before. What excreted products do we humans excrete? Yes, we do excrete urea. And some of the adult amphibians, because adult amphibians will come out of water to live their adult life on land, they will excrete urea. And remember, urea is less toxic, but animals have to spend more energy to produce urea from ammonia, because ammonia is produced and then with the availability of enzymes, ammonia is converted to urea. Therefore, animals have to spend more energy to produce urea. Some marine fishes, like sharks, also excrete urea, which they use for osmoregulation. Now, sharks actively regulate their internal salinity. You know, sharks live in the sea, so the seawater has a lot of salt in it. It's saline. So sharks actively regulate their internal salinity to match the salinity of the outside environment. Therefore, now their waste product is urea, and they use this waste product to maintain osmotic balance as well. Right? After a shark dies, children, what happens is the urea in its body converts into a foul-smelling toxic ammonia. Okay, this is a new fact that I learned as well recently. When a shark dies, because a shark uses its excretory product urea for its osmoregulation as well. And when a shark dies, what happens? It's in the body fluid. The urea in its body will convert into toxic, foul-smelling, bad-smelling ammonia once again. Okay, so I hope you're filling the table with the examples and the types, toxicity, and the energy that is needed for production. Let's move on to uric acid. Now, you might have seen a lot of geckos. Usually, you can find geckos, snails in your houses. Yeah, so some terrestrial animals like birds, reptiles, land snails, and insects. For them, the main excretory product is urea. I'm pretty sure you all have seen these kind of things. Uric acid is relatively non-toxic and generally insoluble in water. Therefore, it is excreted as a semi-solid with very little amount of water. Okay, so most of the reptiles, birds, and land snails, they will excrete uric acid as their main excretory product. Okay, so for them, production of uric acid requires even more energy than urea production because ammonia has to be converted to uric acid, but it is relatively non-toxic when compared to urea and ammonia. Ammonia is very toxic, that is why it has to have a big aquatic environment around it for the organisms to excrete it. Okay, you can see this is a bird and a land snail and a gecko, and most of these reptiles, insects and land snails, they excrete uric acid. So here we are, we have a summary of all of the characteristics of these nitrogenous end products, ammonia, urea, and uric acid. The type of environment, the examples, the toxicity, and the energy required for production. Sometimes, children, there will be an essay for you. You might have to describe the relationship between the nitrogenous excretory products and the environment of the organisms. So you can use this entire table and make a nice essay to describe the relationship. You can say who are the organisms that excrete ammonia and why, how is the toxicity and the energy, and urea and uric acid. So fill the table. In our body, urine is formed, and urea, uric acid, creatinine, water are excreted from the body because we have our kidney and liver produces urea, right? It's not the kidney that produces our urea, it's the liver. Liver produces the urea, and urine is concentrated along the kidney tubules, and creatinine also is from the breakdown of bile. So in our body, yes, urea is the main product, but after that, there can be other excreted products as well. All right. Then that finishes the first part. Then we have the learning outcomes of the second part of our lesson. List the different types of excretory structures found in animals with examples. It has to be with children here. There's a small spelling mistake. Identify and name the different excretory structures using diagrams or charts. So you have to write down the different excretory structures found in animals with their examples and identify the structures. All right. So I have given in your tute the table with the excretory structure and the example. Also, I have given you pictures of the different structures and special points to remember. All right, so I want you to fill those and write down the answers. Okay, now The diversity the diversity of excretory structures of animals. You can see there are a lot of excretory structures with evolution, children. Different phylums have different excretory structures. So most primitive, most simple organisms excrete through their body surface. Then with with the different phylums, some of them have flame cells, nephridia, malphigian tubules, green glands or antennal glands, sweat glands, salt glands, and almost all the vertebrates, they have kidneys. So we have kidneys for our excretion and osmoregulation. Then let's move on to the first type of excretory structure, that is the body surface. Now, cells of some animals which are in direct contact with the environment, they will eliminate their excretory products by diffusion. Right, so the animals, for example, cnidarians or coelenterata, right, hydra, jellyfish, coral polyp, those organisms, they are in direct contact with the environment, and therefore they will eliminate the excretory products by diffusion. So give me 1 second. Okay, sorry if there are disturbances from the outside. So body surface, you can see I've given you pictures of hydra, of coral polyp, of sea anemone, jellyfish. These organisms, they are directly in the marine, directly in contact with the environment. So they don't have to have very developed structures. They can simply eliminate the excretory products by diffusion. Then the flatworm, Platyhelminthes, phylum Platyhelminthes, right? Planaria, you have given you a picture of planaria, and they use flame cells, specialized excretory cells connected to a network of tubules which open to outside of, yeah, sometimes it's called photonephridia as well. So flame cells, we'll use the term flame cells here because it's given in the resource book as flame cells. You can see they have cilia, then the interstitial fluid, there is a cap cell, tubule cell that makes up the flame bulb, and there's an opening in the body wall to excrete the end products of excretion. Okay, so you have to remember the phylums of these animals. So flatworm is a free living organism that has a protrudible pharynx. I'm correct. So they have flame cells. Nephridia. They are multicellular tubular structures. One end of the tubule is open to open to the coelom, while the other end opens to outside. Okay, so nephridia, the ones who have nephridia, like the annelids, they are multicellular. They are multicellular and they have these tubular structures. Remember, in nephridia, one end is open to the coelom of the body and the other end to the outside. Okay, so children, listen and concentrate. I know there's a lot, even in one part of animal form and function, even in one section of this lesson, there's a lot that you have to remember. The diagrams, they have told that fine structures are not necessary, but it's better to remember these diagrams as well. So in annelids, okay, slow down. In annelids, I will go through this once again as a short note before I go to the next part. Now, the ones who have nephridia, like annelids, they are multicellular tubular structures. One end of the tubule is open to the xylem, and the other end, there is an internal opening here and an external opening. One is opening to the xylem, and the other is open to the outside, right? Okay, so after completing the excretory structures, I will go from the beginning and summarize everything that I have said. Then the other one is Malpighian tubules. These are extensive blind end tubules, open at the end, immersed in hemolymph, and opens into the digestive tract. So you have learned about, if you have done circulatory system, you have learned about hemolymph, that some organisms, they have hemolymph in their body, right? And so that their interstitial fluid and blood vessels are not separated. So those kind of insects and terrestrial arthropods, they have Malpighian tubules. You can see there is a blind end open at one end, and these are the Malpighian tubules. Salt, water, and nitrogenous wastes will come into the digestive tract, and through to the anus, the feces and urine will be moved. Okay, yes, you all have done. All right. Green glands or antennal glands. Now, in crustaceans, most of the animals like crabs, prawns, especially, you have learned about the phylums. So two large glands found ventrally in the head and anterior to the esophagus. Here, the green gland is found in the prawn, and here also I've given several images of the green gland. These are found in especially crustaceans, ventrally in the head and anterior to in front to the esophagus. So green glands, or they are also called as antennal glands, found in crustaceans. Then sweat glands. We have sweat glands, especially in the human skin. They are coiled tubular glands situated in the dermis and connected to a sweat duct that opens as a pore. In your body, you have, even though we can't see, we have pores on our skin surface. So under the skin, there is a sweat gland and a hair follicle, and the hair follicle will come out through the pore. The sweat gland is connected to it, right? So that is how sweat is secreted out of the body. That is also another excretory structure found in organisms, especially in humans. Okay, if you have any questions, please. Okay. Do they ask to describe each of these structures? They won't ask you to describe as in an essay or anything, but they might ask you to write. Now they might ask you in an MCQ. They will give you the structure and ask you the example of the organism that has the particular excretory structure. For example, they will give you a picture of a Malpighian tubule, and yeah, because they want you to know the different excretory structures. I told you they want you to know in diagrams and maps like that. Sweat glands, I'm sure it's not only humans that have sweat glands. I will make sure to tell you about it. Okay, so you will learn about the skin in future lessons. Yes, yeah, you will learn about skin. So in there, they will teach you all of these parts, right, about the sweat gland, hair follicles, and then for now, sweat is not toxic, but it is also excreted from the body because it is not necessary to keep it inside our body, right? So it is an excretory product. Um, so I don't think, but there's one thing that can happen with sweat. Children, sweat can get clogged in our body. It is also removed as a way to get rid of heat, right? Temperature regulation, also in temperature regulation, sweat glands are very important. And when we talk about excretion, they are not necessary to be inside our body. So sweat will be removed from the body. And some of it. Salt glands, paired glands found near the eyes to excrete excess salts. These are especially found in the marine birds and marine reptiles. Okay, the salt gland is found with a salt duct, a salt gland duct. In most of the marine birds and marine reptiles, they have salt glands. But one thing I want you to remember, birds are vertebrates, and all vertebrates, they have the kidney is the main excretory organ. So especially for marine birds and marine reptiles, they have salt glands. But if they ask you the main excretory structure of marine birds, don't write salt glands. It has to be the kidney, right? They have, additionally, they have salt glands to excrete the excess salts. Okay. So then we have the kidney, the major excretory and osmoregulatory organs of all vertebrates. I will tell if kingfishers are born to marine birds. I will tell you throughout the lesson. All right, so before going further into labeling the parts, I will go to the beginning once again. I'll just refresh what we have learned till now. All right, so we learned the definition for osmoregulation. We learned that it was a process by which organisms control the solute concentrations and water balance within the body. And we learned that simple unicellular organisms like amoeba or paramecium, they have contractile vacuoles for osmoregulation. And excretion was the method of removing nitrogenous metabolite and also other metabolic waste products from the body. Okay. We also learned that deification is not considered under excretion, that deification does not involve metabolism of undigested food, therefore we do not take it under excretion. Then we learned the importance of osmoregulation and excretion on a greater scale. So we learned that excess weak acids or weak bases are not important, is not good to the body, as they may alter the pH and will cause even the denaturation of proteins. So in order to maintain a constant internal environment, in order to maintain homeostasis, we have to have our concentration of solutes and water balance within favorable limits. All right, and then we talked about the metabolic substrates, carbohydrates, fats, proteins, and nucleic acids. Then we said the factors, we talked about the factors on which these excretory products of the substrates can vary. We said the excretory products can vary on the chemical structure and composition, right? The availability of enzymes, the oxygen availability, and the habitat in which the organisms live. Then we talked of the metabolism of carbohydrate, fat, protein, and amino acids. We said carbohydrates, if they are metabolized, there are two methods, aerobic and anaerobic. In aerobic, carbon dioxide and water are produced. In anaerobic, it's lactic acid that is produced. In fats, they can't do anaerobic metabolism, so it's always aerobic, and they will produce carbon dioxide and water. Proteins, they have amine groups. They will produce ammonia, nitrogenous base, uh, nucleic acids, they have nitrogenous bases, so they will also produce ammonia. And then depending on the availability of enzymes and the habitat in which the organism live, the excretory product will vary from either ammonia, the nitrogenous excretory product will vary from either ammonia, urea, or uric acid. Then only we came to the type of environment and the organisms that will excrete the different excretory products. So ammonia, we learned that it is highly toxic and needs less energy to be produced. But because it is highly toxic, only the aquatic animals that have ready access to water can excrete ammonia as the excretory product, like bony fish, tadpoles, and aquatic invertebrates. Then for urea, we learned that mostly terrestrial organisms excrete urea. All right, and so for them, especially, and also sharks excrete urea. We said mammals and most of the adult amphibians and especially sharks excrete urea. And urea is less toxic than ammonia, but more energy is needed to produce urea. And when it's about uric acids, we learned that they are not soluble in water, and uric acid is relatively non-toxic. We learned that uric acid takes the highest energy for production. All right, and most of the birds and reptiles, they excrete uric acid. Then we came to the excretory structures of different phylums of an organisms. We said cnidarians or coelentrates, they excrete through the body surface, right, through diffusion. And flame cells or protonephridia are found in planaria or flatworms. And nephridia are found in annelids. Okay, in the next future slides, we will be marking the parts, right, just so that you can remember them. And Malpighian tubules are found in especially arthropods. It's much better to remember a picture, children, for each of these parts. All right, so if it is body surface, you can remember a jellyfish or a sea anemone. If it's flame cells, remember the planaria, flatworm. Nephridia, you can remember the earthworm. Malpighian tubules, remember a cockroach. Green glands or adrenal glands, you can remember a crab, right? And sweat glands, humans, we have our sweat glands. I will make sure to tell you which other organisms have sweat glands other than, I'm sure there are other organisms, humans. Then salt glands are found in marine birds and reptiles, but I also told you that since all vertebrates have their major excretory organ as the kidney, you can't write salt glands as the major excretory organ of marine birds as well. Right? Because once when I was doing my A-levels, I got it wrong. So I remember that very well. I wrote salt glands as the main excretory product, but the correct answer was kidney. All right. Then let's move on. Now I heard that I was too fast before. Is that okay now? Or if there's anything I need to explain again, please let me know. I'm sorry if I was too fast. All right. So we'll move on. Now this is a real picture of the kidney. All right, the one that I've given you, the third picture in the slide, it's a real picture. Thank you. All right. Then there's a lot of learning outcomes in the last section. You have to know the parts of the human. You know, it's the human part that we're coming to all. This time we were talking about all the organisms. Right now we are going to concentrate on us, on humans, our structures, and how urine is formed in our body. All right, so we're going to learn about the kidney, then deeper into the kidney, the microscopic structures, the nephrons. After that, how hormones are involved in regulation of our pressure in the body, in the blood, and the diseases that we can have in our urinary system. All in all, we're going to learn about the human urinary system, right? So this lesson is important, children, because I have not seen a lot of MCQs and questions, but in each year, as I've gone through the past papers, I've seen that in each year there is one MCQ. I'm sure you all know the structure of the MCQ paper. It starts with the first lesson and goes until the end. It goes in order. All the questions in your MCQ paper for A level goes in order of your chapters. So a lot of your questions will be from the first part, that is the big resource book, the grade 12 resource book. We'll have a lot of questions for you, and then anyway we will have questions from microbiology, molecular genetics as well. Okay. I know you missed some presentations, some slides, because it was a big lesson. So at the end of the lesson, I'll make sure to send you all a PDF to your emails, a PDF of this presentation, so you can download it and write down whatever that you have missed. Right? Just want you all to make a complete note. Children don't have so many notes here and there, right? Because then at the end you don't know how or from which note you have to study. All right, just it's never too late. Even today you can start. Just organize everything, have a good note, right? Because a good note means a good theory, and if you have a good theory in biology, it's very easy. Just have to write down what they are expecting from you, right? So that is why they have given the learning outcomes as well. So let's start. The human urinary system. Okay, so the parts of the human urinary system, we have 2 kidneys, 2 ureters, 1 urinary bladder, and 1 urethra. All right, 2 kidneys. Now, when I tell this, imagine the structure in your mind: 2 kidneys, 2 ureters, then both of the ureters come and join to the urinary bladder, and after the urinary bladder, there is a urethra. All right, so wait, did I miss the structures, the labeling parts? Well, okay, in the tute, I have given you to label the structures, label the following parts and write the names of the structure. And I've given a place for special points, right? So for that, I will send you that part. Okay, I will send you a small PDF with that page. I will write the special points you have to remember as well in each of the excretory structures. All right, anyways, it's in the presentation, but since you have to know what the special points are, I'll just make sure to send you a PDF of that as well. Somehow or the other, I just want you all to have a good note, children. All right, so now there is a table that I have given you. You have to fill the table, the functions of kidney, ureter, urinary bladder, and the urethra. Sometimes when you are given to mark or label, children, you will miss or you will forget, and you will write ureter for urethra and urethra for ureter. That happens, okay? Even if you are 100% confident, even if you have studied, you will miss, you might miss. So you have to be prepared. You have to mark these again and again, children, and always double check when you're doing something. I have experience when I'm learning, and so before moving on, I'll tell you a few. Always make sure to do past papers. Now, when I finish this lesson, I want you all to find the past paper questions and take a paper, write down the year and the question number, write the topic, osmoregulation and excretion, and write down the question numbers with the years, so that at the end, when you want to study, you know these are the years and these are the questions they have asked. All right. When I did my A-levels, I actually wrote down all the essays from 2000 to 2000 to my year. I wrote down all the questions and all of the essay questions that have been asked from each of the chapters. If you have some free time, please do make your work easy, because biology is not hard. It's about remembering the facts, children, just remembering. So don't make it hard, don't make it complicated. Just simply study. Don't procrastinate. Don't say I'll do it tomorrow, or I have so many days before the exam, so I'll do it till the end. No, that won't take you anywhere. Just regularly study. All right, keep a note, keep a good theory, do past papers, especially time the past papers and do them. Then it won't be a big deal for you. Trust me, exam is easy. Biology is very easy. You can always get above 40 for the MCQ if you are thorough with your resource book. They won't ask you anything out of the resource book. It's not that hard. All right. So back to the lesson. Now, kidney, the main function, produce urine to excrete waste products and while maintaining osmotic balance and acid-base balance. So I've told you that in humans or in vertebrates, the main excretory organ is the kidney. So to maintain the osmotic balance and acid-base balance, the kidney is very important. Also to produce urine, right? That is the way that we can remove the nitrogenous excretory products from our body. Then the ureter receives urine from the kidney and sends it to the bladder. Urinary bladder will temporarily store the urine. Then urethra will provide the passage through which urine stored in the bladder leaves the body. All right, so children, just remember it like a picture. Just imagine the urinary system, two kidneys, two ureters, urinary bladder and the urethra. Kidney produces urine, right? Just also remember that it maintains osmotic balance and acid-base balance. Then the urine produced in the kidney will go into the ureters, and ureters will take it to the urinary bladder. Urinary bladder will temporarily store the urine, and when it's time to pass, we can control there's a sphincter at the urethra. We can control the passing out of urine, and when we want to pass out, we will pass urine through the urethra. So it's easy to remember it as a process with a picture. We always remember things much better in picture mode. So imagine the system, the urinary system, and remember the functions. All right. So write down the functions as much as you can. I will share the PDFs. You can complete the rest. All right. Otherwise, this lecture could go on for long because we have not yet come to the process of urine formation. That is a bit lengthy, and I've made sure to break it into slides and explain with colors, because in the resource book it's all in together. It's hard to read, and I'm sure we want to read if there's a lot like that, right? So I've made it in short form. You'll see. Moving on, location of the kidneys. Now this is also important. When you're asked to write the location of the kidneys, children, remember all of these 5 points. All right, just remember it as a picture once again. On the posterior abdominal wall, on either side of the vertebral column, behind the peritoneum, below the diaphragm, right kidney is slightly lower than the left. Remember that part, and then you will get full marks if you were asked to write the location of the kidneys. Okay, that is how I remember. Just imagine, just draw a little sketch and mark on the posterior the abdominal wall, right? We have the thoracic cavity and the abdominal cavity in our body. Thoracic cavity is the chest, abdominal cavity is below the diaphragm, the part of the body. And so on the posterior abdominal wall, And on either side of the vertebral column, you can see the vertebral column from behind. And on either side of the vertebral column, behind the peritoneum, okay, I will explain, and below the diaphragm. So now I will describe the parts of the body. Now the chest area is called the thorax. This is the thoracic cavity where you have the ribs. It's the thoracic cavity. And above the diaphragm, I'm sure you all know the diaphragm in the respiratory system. Above the diaphragm, it's the thoracic cavity. Below the diaphragm, it's the abdominal cavity. All right. So in the abdominal cavity, in the abdominal wall, the posterior, in the back side, if you touch your back from your hand, like in here in the picture that I've given, in the posterior abdominal wall, and you know the vertebral column of our body in the skeletal system, the backbone, on either side of the backbone, behind the peritoneum here, and below the diaphragm, right kidney is slightly lower than the left. So make sure that you all describe all of these 5 points. Just like I told you, if you write the 5 points, you can get full marks, right? These words, just remember as in a picture. It will be really helpful for you all, children. Okay, peritoneum, it's a membrane, children, that is lining the abdominal cavity and covering the abdominal organs. You know the organs in the abdomen, right? The stomach and all these organs in the abdomen. So the peritoneum is like a membrane that is covering those abdominal organs. All right. So behind the peritoneum only, behind the layer, behind the membrane that is covering the abdominal organs, and below the diaphragm, you will have the kidneys. Right kidney is slightly lower than the left. When you're drawing also, make sure to draw the right kidney a little bit lower than the left. And how do you remember left and right? Children, I hope you all know in the body, how do you remember which side is left and which side is right? Simply if I tell, your right side will be the left side of the body of whichever structure that you are asked to label, and your left side will be the right side of the structure. Let's say you are given the structure of a heart and you are to label that structure. Your right hand side will be the left of the structure of the heart, and your left will be its right. Just imagine someone with a heart is looking at you, so his left will be your right. Just try to imagine that. Okay, if you didn't get it, I'll explain again. So remember these 5 points. All right. Then the blood supply to the kidney. Now we're going on to the human urinary system, the urine formation and things, children. So it's going to be a little bit lengthy, but please do listen. Okay. So the blood supply, the kidneys receive blood from the aorta, while the renal arteries and renal veins return blood to inferior vena cava. Okay, kidneys will receive blood from the aorta. You know the heart pumps blood, and blood is pumped to the body from the aorta. So through the renal arteries, kidneys will receive blood, and after the blood is used up, that blood will be returned to the renal veins, and renal veins will take that blood into the inferior vena cava. That will again take the used deoxygenated blood into the heart. So I hope it's clear. Initially, aorta will bring blood, and from aorta, renal arteries will take blood to the kidneys. And from the kidneys, renal veins will take the deoxygenated blood into the inferior vena cava. All right. Okay. So any questions? None of you all have questions? Or is the session, was I too fast? Tell me if there's anything I need to explain. All right. Then moving on to the gross structure of the kidney. Now this too, they will ask you children to describe this. They might ask you in an essay or in a short note to explain the gross structure of the kidney. Okay. So remember now I've given in points the gross structure of the kidney. It's a bean-shaped organ. The average length, you don't have to remember, but I just included that too. It's about 10 to 13 centimeters long, and the breadth is approximately about 6 centimeters. This is a real kidney, a longitudinal section. So when they ask you the gross structure, you don't have to explain all about the nephron. No, you just have to explain the outside structure, the structure that you can see. Not, you don't have to go into detail. So it's a bean-shaped organ held in position by a mass of fat, both surrounded by a fibrous connective tissue. And in the longitudinal section of the kidney, you can see three areas to your naked eye: the outer fibrous capsule, renal cortex, and the inner renal medulla. In the structure of the kidney, in the next slide, I have given you the parts of the kidney, and in the tute as well, I have given you to label the parts. So you can label looking at that diagram that I've given. Cortex and medulla. So here is the renal cortex, and the renal medulla are supplied with blood vessels and tightly packed with excretory tubules. The renal cortex mass of fat. Yes, so I said the kidney is held in position by a mass of fat. For it to stay in position, there is an amount of fat, a little bulge, I think, that is keeping it in position. On top of the kidneys, you will find the adrenal glands. So for the kidneys to be in position, there's a little bit of fat that is holding it up. A fat, bit of something like a little muscle, not muscle, but it's a bit of fat that is holding the kidney in place. Yes, I will send the presentation after the session. I'll send a PDF, but it is much better if you can listen, because the presentation, it's long, it's lengthy, and I hope you all listen. Okay, and moving on. Medulla is composed of renal pyramid, and renal cortex is granulated due to the presence of glomeruli. There are structures called glomeruli, and so the renal cortex looks granulated with granules. It looks like it's filled with granules. Medulla is composed of renal pyramids that have a striated appearance, because in the medulla they have renal pyramids in the structure of a pyramid, right? It's not exactly a pyramid, but that's how they have described it. So in the renal medulla, they have renal pyramids, these structures that have a striate, gives it a striated appearance. Apices of the pyramids, these apices project into the renal pelvis through renal papillae. All right, so the renal pelvis is found in the middle here, and that is where the ureter starts out of each kidney. This is the longitudinal section. And the other thing I want you all to remember, children, is sometimes they will give you a structure like this and ask you to label, ask you to tell what that structure is. If you just say it's a human kidney, it's not correct, right? That you can see is a longitudinal section or an LS of a human kidney. So you will have to write that it is a longitudinal section. If you just write a human kidney, that is not very accurate. All right. So there can be, when you're, now I am sure you must have learned TS, transverse section, LS, longitudinal section, when you are doing dicots and monocots. Yeah, so here also, if they asked you to name the structure, it's a longitudinal section of a human kidney. Okay. So that is the gross structure of the kidney. Just remember, there is a cortex, there is a medulla, then they are supplied with blood vessels, tightly packed with excretory tubules. Cortex is granulated because it has glomeruli. Medulla has these renal pyramids that gives it a straightened appearance. And the apices, the tips of these pyramids are projected into the pelvis through their renal papillae. And the ureter starts from the pelvis. All right. Moving on. And the other point, renal artery and renal vein pass through the pelvis. You can see renal artery and renal vein are passing through the pelvis. Then the structure of the nephron. So then now only we are coming into the deep part of the urinary system of the kidney, the deep part of the kidney. All this time we were talking about the gross structure, what you can see from outer surface. Now we're going into the kidney and we're talking about the structure that forms a kidney. All right. So nephron is the structural and functional unit of the kidney. Children, can you tell me what is the structural and functional unit of the brain? Just asking a side question. What is the structural and functional unit of the brain? I want separately the answers. If you can, please put in the chat. The structural and functional unit. Okay, I want to see your answers. A lot of you. I want you all to. Okay, just remember that in the brain, you have learned this in O levels. Okay. Yeah, all of you, a lot of you have said neuron and some of you have said cerebrum. I will clear it for you. The structural unit of the brain is the neuron, but the functional unit, yes, one of you have given the correct answer. Structural unit is the neuron. Functional unit is the reflex arc. I hope you remember the reflex arc. If you touch a hot object, you quickly take your hand off. That reflex arc and the neuron are this functional and structural units respectively. But in the kidney, both the structural and functional units is the nephron. That is what I wanted you all to know. In the brain, structural unit is the nephron, functional unit is different, it's the reflex arc. But in the kidney, both structural and functional units are the nephron. Right? So there are so many, over millions of nephrons are there in each kidney, and two types of nephrons I am showing the question. In the tute I have asked you what are the two types of nephrons: cortical nephrons and juxtamedullary nephrons. Okay, cortical and juxtamedullary. Cortical nephrons reach only a very short distance into the medulla. Okay, cortical nephrons reach a very short distance, but juxtamedullary nephrons will extend deep into the medulla. If you look in this picture, you can see a cortical nephron. It hasn't gone very deep, but this juxtamedullary nephron has gone deep into the renal medulla. Okay. Yes, it's the nervous system that has reflex arc. That is true. I was just asking you if in the nervous system, what is the structure and functional unit, because those two are separate, right? A lot of you said that neuron was the structural and functional unit. I wanted to clear that out as well. Neuron is just the structural unit and reflex arc is the functional unit. That is true. Here, when we're talking about the kidney, both structural and functional units are the nephron. That is what I wanted you all to know. Okay, and back to the lesson. Remember that a majority of the nephrons are cortical nephrons, so the nephrons don't go very deep into the medulla. Okay, nephron is the structural and functional unit, and there are millions of nephrons that make up each kidney. Okay, all right. Parts of a nephron. Now you can imagine how big animal form and function lesson would be, right? You have done digestive, respiratory, immunity systems, and circulatory systems as well, I think. And you have excretory here. There is nervous, yeah. And so it's a big lesson. You just have to remember everything like a story with the correct, accurate words, children. But if you study, then if you study regularly, it won't be that hard. Otherwise, you will have to spend the entire day studying because you procrastinated, and then it would be hard remembering all of that on the day of the exams. Just remember as you learn and as you hear. Okay, children, do you need about a 10-minute break at about 3? Because we have someone who is about to, yes. Okay, I will give you at 3, not now. I will give you a little 10 minutes break. You can refresh and please come back to the lesson. Be prepared just like you were all this time. All right, because it's going to be a little bit lengthy. I'm going to give you a little break. Okay, so parts of a nephron. Now we're going to study in detail about the nephron before we're going into urine formation. A nephron consists of a single long tubule and a ball of capillaries called the glomerulus. Okay, so nephron has a single long tubule and a ball of capillaries. Together you call it the glomerulus. The tube is closed at one end, forming Bowman's capsule, which surrounds the glomerulus. The other end of the tubule joins with the collecting duct. So collecting duct is where urine is collected at the end. So one end of the tubule of this nephron is connected to the collecting duct, and the closed end is connected to the Bowman's capsule, which is surrounding the ball of capillaries, or which is surrounding the glomerulus. You might get to write about the parts of a nephron as well. Okay, so make sure to draw a diagram whenever in an essay. It's not always necessary, but it doesn't matter if you are asked to describe some structure. If it's a gross structure, right, let it be the structure of the kidney or structure of a nephron or structure of the cerebrum whatsoever. Make sure that you draw the structure and label the parts, because I'm sure it will add more value to your essay. Even if it's about the parts of a nephron, you can draw a little structure of the Bowman's capsule, the tubules and the collecting duct. Yeah, and it would be great. So the tubule of the nephron consists of Bowman's capsule, proximal convoluted tubule, loop of Henle, and distal convoluted tubule. Okay, Bowman's capsule, that is what we talked of, which surrounds the glomerulus. The PCT, that's how we use in short form, but you can't write short form in the exam paper. All right. If it's an essay, children, let's say it's hard, right, to always keep writing proximal convoluted tubule, distal convoluted tubule. I understand, but if you're going to write short forms, first you have to write the long term, and within brackets you can say if it's proximal convoluted tubule, you can write that, and you can write within brackets PCT. Then as you are continuing, you can use the 3 letters PCT. But I like if you write the entire term when you're writing the essay, because the short terms are not much appreciated in the paper. All right. Then, so the tubule has Bowman's capsule, PCT, loop of Henle, and the DCT or distal convoluted tubule. So nephron has a long single tube and a ball of capillaries. This long single tube has 2 ends. One is closed, surrounding the Bowman's capsule, and the other joins with the collecting duct. This tubule has 4 parts: Bowman's capsule, PCT, loop of Henle, and the distal convoluted tubule. Is that clear up to here? You let me know if everything is clear, or if I have to redo or tell anything. Do you understand everything? Can you please put on the chat, children? Okay, I'm glad if it's clear to you all. Nephron is naked. I don't know what nephron is. Okay, I'm glad it's clear to you all. Okay, let's move on. Now, Bowman's capsule. So it's also called the glomerular capsule because it surrounds the glomerulus. Okay, it's like a capsule. You know, capsules surround something, right? So it's like a capsule surrounding the glomerulus. We call it the Bowman's capsule. It must be a name of the person who found it, or the glomerular capsule. Expanded and closed end of the tubular structure of the nephron. So glomerular capsule is the expanded and closed end of the tubule of the nephron. All the parts of the nephron, okay, this picture is not very clear, but this is what you have in your textbook. I took it from the resource book. This is one nephron. You have millions of such nephrons that make up a kidney, right? So the afferent, efferent, we're going to learn parts of the each and every part, and I'm sure there are pictures of nephron in future slides. We'll see. So this is one entire nephron, okay, and I will try to find a enlarged picture. So here, expand and closed end, it's cup shaped and it's double walled. There is an inner layer and an outer layer. Okay, the inner layer of Bowman's capsule has a single layer of flattened epithelial cells. You have learned in the first part of animal form and function, when you were starting animal form and function, you learned about the tissues, the epithelial tissues, what are the different kinds, then the connective, so on. So here in the inner layer of Bowman's capsule, you have flattened epithelial cells that are specialized for filtration. Okay, because glomerular capsule or Bowman's capsule is involved in filtration of blood, because a process called ultrafiltration is done in the Bowman's capsule. Okay, so I'm asked to explain about the location and gross structure of the kidney, how to answer a structured question. Yes, an essay question. So yeah, I will, at the end of the lesson, I have put, at the end of the presentation, I have put some essay questions, and this is included, the gross structure of the kidney. I will make sure to tell you how to answer an essay and structured question, all right? Okay, so moving on, now the two layers of the Bowman's capsule. In the inner layer, remember, flattened epithelial cells are there for filtration, and in the outer layer, it has simple squamous epithelium. Okay, the simplest form of epithelium, simple squamous epithelium in the outer layer, and inner layer, flattened epithelial cells, especially for filtration, because ultrafiltration, the first process of urine formation, takes place in the Bowman's capsule. Okay. Then space between inner and outer layers is called the capsular space. So inner layer and outer layer, the space is called the capsular space, that is responsible for receiving the glomerular filtrate. Now this is the glomerulus. From the glomerulus, some particles will filter into the space of the Bowman's capsule. So the space between inner and outer layers of the capsular space is called the capsular space, that will receive the filtrate from the glomerulus. All right, the glomerular filtrate passes through three major tubular regions of the nephron: the PCT, loop of Hindley, and the DCT, distal convoluted tubule. So we will be talking about the glomerulus in the next slide. Before that, remember, this space is called the capsular space, that is going to receive the filtrate from the glomerulus. Okay. Then the glomerulus, a ball of capillaries surrounded by the Bowman's capsule. Nephron is supplied with blood from the afferent arteriole. I have colored afferent and efferent, the first two letters in red. That is for you all to remember the words correctly. Inner and outer wall of the Bowman's capsule. Okay, one sec. Okay, fine, here. So this is the inner wall. Okay, no, sorry, this is the inner wall here, and okay, fine, the capsular space is this. So if you take the Bowman's capsule, you can see the inner wall that is facing the capsular space, and the outer wall is to the outside, to the side of the glomerulus. So the filtrate of the glomerulus will come into the capsular space, which is covered by the inner and outer walls. Inner wall is towards the capsular space. Okay, here. So back again to glomerulus. Now I said I colored afferent and efferent in the first two letters because otherwise you will mix it up. Okay, so in the glomerulus, nephron is first supplied with blood from the afferent arteriole. Arteriole is even smaller than the artery. All right, so it's the aorta, then the artery, and then a mini version of an artery is an arteriole. So in the glomerulus, the one that brings blood to the glomerulus is an afferent arteriole. It's called an afferent arteriole. And remember, it's afferent, and the one that is taking blood away from the glomerulus is called the efferent arteriole. Okay, that has a E in it. So remember, it's in alphabetical order. The one that brings is A, the one that goes out of the glomerulus is E. That way, it's easy to remember. Okay, so afferent arteriole has a larger diameter than the efferent one. You can see very well in the picture, right? This one has a larger diameter, and efferent arteriole has only a little diameter. Why? Because when blood when there is a large diameter, a lot of blood can flow through this afferent arteriole, and it can go into the glomerulus. And when the blood is trying to go out through the efferent arteriole, the diameter is restricted. So it comes with a big pressure, but it can't go out in the same pressure. So a big pressure will build up inside the glomerulus. Inside the ball of capillaries, there will be a big pressure because there was a larger diameter, so a lot of blood came, but all that blood can't go at once. So inside the glomerulus, there will be a larger pressure that is generated. They want the pressure to be generated because the particles have to go into the capsular space. So that is a specialization that we find in the glomerulus. See how our body itself is adapted to even the minute, the tiny structures are very well adapted to carry out the functions of our body, right? The afferent arteriole has a higher diameter. Efferent has a lower diameter compared to afferent. Why? To increase the pressure of the glomerulus so that blood can be filtered and the other substances can go into the capsular space as the glomerular filtrate. Okay. Then the efferent arteriole forms two capillary networks, one from the peritubular capillaries. So in the picture, they have given the peritubular capillaries that are surrounding the PCT and DCT, and the other network is the vasa recta that is in the bottom that surrounds the loop of Hindley. Okay, the efferent arteriole that had the smaller diameter and one that brought blood out of the glomerulus forms into two capillary networks, one from the one called peritubular capillaries that surrounds the PCT and DCT, and one called vasa recta that surrounds the loop of Hindley. All right. So all of this is the structure of the nephron, remember. Then the proximal convoluted tubule, PCT. So they have given you all of these parts separately, PCT, loop of Henle, and then to the urine formation. All right, so proximal convoluted tubule, this is comparatively longer and wider than the distal convoluted tubule. Okay, lined by simple epithelium, just like the Bowman's capsule, specialized, but here it is not specialized for filtration, but for selective reabsorption. What do you mean selective reabsorption? Some things missed, some of the substances might have missed and gone into the glomerular filtrate, because the ones that are going from blood into the glomerular, into the capsular space, are the ones that are going to be removed with urine. But some important substances like glucose or amino acids, they need to be taken back into the body, reabsorbed into the body selectively. So that is why selective reabsorption is done by the proximal convoluted tubule. Okay, nutrients, ions, and water from the glomerular filtrate will be taken back into the blood. Right. Loop of Henle. This is the part I've shown in the picture, the loop of Henle. It is a U-shaped part of the nephron with a descending limb and an ascending limb that is surrounded by simple epithelium. Remember, first it's the descending limb. It will descend first and then it will ascend. Okay, don't mix up those two. Loop of Henle. First, from the PCT, it will come to the descending limb of loop of Henle and then into the ascending limb. Lining of descending limb is specialized for water reabsorption because it allows movement of water, but ascending limb of loop of Henle is impermeable to water. Remember that as well. The descending limb is permeable, it allows water exchange, but the ascending limb, as it starts ascending up to the distal convoluted tubule, it is impermeable to water, which means it doesn't allow water to get reabsorbed. All right. So that is what we were talking. Now, if I just remind you from the last part, we were talking about the human urinary system. So we said the parts of the urinary system were the kidney, the ureters, the urinary bladder, and the urethra. Then we discussed their functions, right? Then we said we talked about the blood supply, the location of the kidneys first, and the blood supply of the kidney, and then we talked about the gross structure. I will tell you how to write for an essay and for structured. There isn't a particular number, about 6. If you have to draw, make sure you draw about 6 medulla in a kidney, right? If you are asked to draw. And then the gross structure of the kidney and the parts of the kidney, right? If you're asked to write about the gross structure, make sure that you draw a kidney and label its parts. Then we talked that nephron is a structural and functional unit of the kidney. The 2 types of nephrons, cortical and juxtamedullary. And we said cortical were the major out of them. Then the parts of the nephron, it's a single tubule with a wall of capillaries called glomerulus. Parts of the tubule, we talked about the Bowman's capsule or glomerular capsule, the glomerulus and PCT and loop of Henle. The descending limb comes first. It's permeable to water, but the ascending limb is not. It's impermeable. Next, we have to talk about the DCT and urine formation and the role of hormones and the diseases. All right, so before that, I'll give you a 10-minute break. So please make sure that you come back by 3:12. Till then, I will, there is a friend of yours who is going to entertain you with a song. Yeah, so please come back by 3:12. I'm going to start exactly at 3:12. Okay. Structure of a bone. Structure of a bone isn't given on resource book, but they have asked in a paper. Structure of which bone, darling? Can you tell me exactly which one? If you tell me which structure, I'll be able to tell you if you have to write it or not. Under connective tissues. Okay, can you just write the question that is given in the paper? If you have the paper, just write down the question so I can check it and I can give you the answer possible here. Okay, for the rest of you at 3:12, please join back.
Okay, thank you, Miss. Now I would like to invite my friend Samanali Amana Kandala to sing a song, and she was the most popular singer in Sri Lanka at 2014. Thank you.
Thank you, Darshika and Parmi teacher for inviting me to sing a song. So, as I have cough, if I cough in the middle of the song, I'm so sorry. I apologize, you all. So I sing the song. मैं जान ये पार तू हर जीत भी हार तू कीमत हो कोई तुझे बेइंतहा प्यार दो मैं जान ये पार तू हर जीत भी हार तू कीमत हो कोई तुझे बेइंतहा प्यार दो सारी हदें मेरी अब मैंने तोड़ दी देखकर मुझे पता आभार की बन गए हाँ हंसी बन गए हाँ नमी बन गए तुम मेरे आसमान मेरे ज़मीं बन गए हाँ क्या खूब रब ने किया, बिन मांगे इतना दिया, वरना है मिलता कहा, हम काफिरों को खुदा। क्या खूब रब ने किया, बिन मांगे इतना दिया, वरना है मिलता कहा, हम काफिरों को खुदा। हसरते अब मेरी तुमसे है जा मिली, तुम दुआ अब मेरी आखरी बन गए, हाँ हंसी बन गए, हाँ नमी बन गए, तुम मेरे आसमां, मेरी ज़मीं बन गए हो। हाँ Thank you so much.
Thank you, Amma.
Okay, children, we're going to start. At 3:12. So please join back and start concentrating on the lesson. All right. Okay, we're going to start, children. Can you tell me if you all are all here? Because if you miss out, it's not good. So I want you all to listen and go with me till the end of the lesson. So can you please put a yes if you all are here, and so we can know how many of you all are already in the lesson. So far, I found only 12 yeses. Okay. About photosynthesis, if you want a seminar, darling, please talk to Tharushika from online class group. I guess you can direct message her. Then you can talk with her. You can say, yeah, it's much better to talk with the online class, one of the admins, and so we can, if you want, we can arrange another session. All right, so I'm going to start. Okay. Now, we were talking about the loop of Henle, and we came to the distal convoluted tubule. It is lined by simple epithelium, which has been specialized for selective reabsorption of specific ions and water. All right. So the distal convoluted tubule, I told you when we were talking about the proximal convoluted tubule, we said that it was much longer than the DCT. So DCT is a bit shorter, but DCT only leads into the collecting duct. That is where urine is finally formed and at the end. So DCT is also lined by simple epithelium. It also does the same function as PCT, selective reabsorption. Okay. Then 3 main steps of urine formation. I have asked a question, what are the 3 main steps? Ultrafiltration, selective reabsorption, and secretion. Those are the 3 main steps in urine formation. And from the next slides, I have given in your tute a picture of the slide itself, because there's a lot to write and it's quite lengthy. This is where the lengthy part starts, all the processes. All right, and after this, there's not much. This is like the most lengthiest part of the lesson, and this you might get to write an essay. All right, they might ask you an essay, describe the process of urine formation. Then you must explain that there are 3 parts. Just make sure the number is also mentioned, children. Sometimes in an essay, if you write there are 3 main steps in urine formation, for mentioning 3 itself, you might get a point. All right. So if they ask you to write the main steps of urine formation in an essay, you have to write there are 3 main steps: ultrafiltration, selective reabsorption, and secretion. For saying these 3 and for saying the number of steps, you will get 4 points. Okay, it's very easy to obtain points in essays, children. So just know what you have to write when you get something like that. Now, ultrafiltration. Filtration of blood under high pressure into the cavity of Bowman's capsule is called ultrafiltration. This is the definition, and the definitions I have given in purple color, right? Filtration of blood under high pressure into the cavity of the Bowman's capsule. So we know why there is a high pressure. I said because of the afferent and efferent arterioles and their diameters, there will be a high pressure in the glomerulus. And because of this high pressure, blood will be filtered into the capsular space or cavity of the Bowman's capsule. Then Filtration occurs through the capillary walls of glomerulus and inner wall of the Bowman's capsule. Blood capillaries of glomerulus are porous, and cells lining the Bowman's capsule are specialized for filtration of small size molecules and ions. Now, this is important. Sometimes they will ask you to write the differences between a normal blood capillary and capillaries of glomerulus. One point is that the blood capillaries of glomerulus are porous. They have pores or holes through them, right? Small microscopic pores, so that all except large particles will go into the glomerular filtrate. And cells lining the Bowman's capsule are specialized for filtration of small sized molecules and ions. These specializations allow passage of water and small solutes through the blood capillary walls into the Bowman's capsule. So from blood, everything except blood cells, platelets, and large molecules like plasma proteins will go into the Bowman's capsule. Remember, three particles will not go into the Bowman's capsule. What are they? Blood cells, platelets, and plasma proteins. Because they are bigger particles, they won't go into the Bowman's capsule and will remain within the blood. The filtrate in the Bowman's capsule contains salts, amino acids, glucose, vitamins, nitrogenous weights, and other small molecules. So everything except blood cells, platelets, and plasma proteins will go into the glomerular filtrate. So it will have salts, amino acids, glucose, vitamins, and nitrogenous wastes. All right. So if they ask you what are the substances found in the glomerular filtrate, you have to write salts, glucose, etc. So and they might also ask you for a structured question. They might ask you what are the structures that remain in the blood without going into the glomerular filtrate, right? So for that, blood cells, platelets, and plasma proteins are the answer. Okay, so make sure if it is an essay, like I told you, you have to write there are 3 processes. And what are the 3 processes? Then explain each of them. And I will tell you another point in writing essays, children. If you didn't know, please don't write points, okay? Or don't draw tables. They don't give marks for tabular structures, even if your answer is correct. Don't draw tables. To study, drawing a table and studying is all right, but when you're writing an essay, it has to be an essay. Just put a nice introduction. It doesn't have to be literature, right? Just a nice introduction of what you're going to write about. And in the body of the essay you're writing, you have to include all the points. You're not writing an essay, it's not a narrative essay. You just have to write all the points that they're expecting out of you. If you're getting 50 marks for 50 points, then you have to write which means it's all from the resource book, children. And the other thing I want to tell you before I continue with this, in some school papers, if you're doing school papers, make sure that you have the answers for those papers. I know most of you all will try doing school papers, and it's all right if you have the answers, because school papers, they sometimes put a lot of stuff outside from the resource book as well. All right, but the exam paper won't be so hard. Trust me, it won't be that hard. It's at least 96% of it is from the resource book. So you don't have to be scared of that, because sometimes from school papers, they will ask you difficult questions and out of the syllabus questions as well. Just remember, you don't have to be scared of those papers, and only do those papers if you have the answer scripts with you. Okay, if you don't have answers, don't try doing those papers. Now you have 2019 and 2019, 20 and 21 papers you all will have. So you all can do those papers and practice with time. Okay, so Yeah, and then about writing, don't write in point form. Always make sure to write in nice paragraphs with pictures if necessary, with drawings, labeled drawings, so that you can score a lot of points too. Okay, so ultrafiltration is done. Simply the filtration of everything except blood cells, plasma proteins, and platelets into the cavity of the Bowman's capsule. Is it clear up to there, children? Can you please tell me if it's clear? Or if I need to tell or teach you again, just put in the chat. Good. Okay. Please explain it again. I will explain it once, yes, since some of you want to explain again. And it doesn't have to be continuous, Daddy. You don't have to, the only thing I'm asking is don't write in points. Don't put bullets or numbers. Don't use that. Don't draw tables or maps or charts whatsoever. Draw a nice labeled picture. Let's say if you're asked to write the gross structure of a kidney. Now I'll explain it here then. If you're asked to write the gross structure of a kidney, you will write the question. Don't use colors, just use blue or black, and draw the picture if you want. Because if you draw the picture first, then you know what you have to write, right? If it's a kidney, draw the kidney, mark the cortex, medulla, and everything. Then write that it's a longitudinal section of a kidney. Then start. The first paragraph, you will say the location of, here you can draw with the pencil. Doesn't matter. But it's better to mark with the pen. Drawing with the pencil is okay. Yeah, don't draw with the pen. Draw with the pencil, but don't keep rulers. Just you have to draw it freehand. Okay. So I hope there are things that you can take home. One essay time also, children. Now the full paper is 3 hours, right? The second paper. So make sure you take 1 hour and or 1.5 hours of the 3 hours. I don't like to, I mean, just don't take entire 1.5 hours for the structured. Just try to do the structured as fast as you can. Some people will tell you, some teachers will tell you to finish half of the structured paper and then finish the essay and come back to the structured. I have tried that method as well, but it didn't work out for me. I'm not sure if you're fast enough, you can come back to the structures. But I myself, I take a lot of time writing the essay, right? So, and once I tried doing half of the structured paper, and I kept it, and I went to essays, and I started writing the essays, but it took longer. It took a longer time, and then when I came back to the structured essay questions, I didn't have much time, so I thought I'd never do that again. I quickly finished the structured essay part. This is just to write the answers, right? Whatever that you know you have to write. Take 1 hour or 1 hour and 15 minutes for the structured essay, and use the entire remaining time. Break it into 4, because you'll be getting to write 4 essays, right? Break it into 4, and make sure to finish writing the essay within the time for each one. Like if it's 1 hour that you have remaining, take 15 minutes for each essay, and at the end of 15 minutes, keep a space. Keep at least half of a page or 1 page. If you have to write more, keep half of a page or 1 to write. If you remember anything, you can write. So keep that and then move on to the next essay. Like that, you have to finish all the 4 essays, because then you know that you have already done the structured. You just have to check it, right? If you do half of the structure and come to the essay and go back to the structure, what will happen is that you'll be in tension, because you know time is going really fast when you're doing the paper. Okay, trust me, it goes very fast. So, and because you're tensed, right? Not like school papers or papers you're doing at home. So if you do half of the structure and you go to the essay, then what will happen? You know, in your mind, I have to complete that half. That half is remaining. So it will be difficult to focus on the essay as well. So as soon as you can, finish the structured well, answer all the questions. If you want, take like 5 minutes and go through everything. 5 or 10, go through everything that you've written, and then come to the essay and divide the time that you have. You have to take a watch to the exam, definitely. 1.5 pages for an essay is enough, or 2 if you can, because the more you write, you have to write the points, right? If it's ultrafiltration, you have to write the slide, okay? Whatever I've given in the slide, you have to write it. And if you just write about the structure of the kidney and stuff for ultrafiltration, writing 2 or 3 pages is useless, right? So whatever that you have to write under the topic, the exact points, just read the question very, very well, okay? Because once no, I'm going out of the way to tell you these parts about the essays, but that's okay, because these parts are important as well to you all. Now, when you all are writing, thing is, okay, I'll tell that to you all later, but before that, now I was talking about the pages. 1.5 is okay. 1.5. So write what you have to write. Don't now sometimes when they give the questions, especially, they are very tricky. You might miss words. You might not. You won't be reading the question. You just read the first 2 words, and you all will this is the question, and you'll be writing so much, and then when you go back, you will realize this is not what they asked me to write. Okay, they might be specializing some part, okay? So because of that, read the questions very well, right? And write about 1.5 pages or 2 for each of them if you can. It's very good if you can write neatly, write the essay. You don't have to have good handwriting, children. Just have neat handwriting, a little bit big handwriting. It doesn't have to be very huge. But after each question, start a new question with a new paper. All of these I'll tell you at the end as well. So, and the other thing, yes, you can have the recording if you want. I will send it to online class group, so you can take it from the admin. And okay, about the essays and essay writing, we'll talk till the end of the lesson. Now we'll go back to ultrafiltration. I will do this once again, right? Because some of y'all wanted it. Now, ultrafiltration, like I told you, takes place in the Bowman's capsule. The afferent arteriole has a larger diameter than the efferent arteriole. Therefore, when blood comes to the glomerulus from the afferent arteriole, there will be a very large pressure that is generated in the glomerulus because of the difference in the diameters. And because of this pressure, blood will be filtered into the cavity of the Bowman's capsule. This is called ultrafiltration. Ultra because it gets filtered in the microscopic level, so filtration occurs through the capillary walls of glomerulus and inner walls of Bowman's capsule. Now in the glomerulus, the blood capillaries are different, a bit different than normal capillaries. Blood capillaries of glomerulus are porous, which means they have pores, right? And cells lining the Bowman's capsule are specialized for filtration. So because of these specializations, they are porous and they have been specialized for filtration of small substances. All of the small substances, solutes and water will flow from the blood capillary walls into the Bowman's capsule. But large particles like plasma proteins, platelets and blood cells, because they are large in size, they can't go into the glomerular, into the Bowman's capsule through the pores. Therefore, they will remain, and everything except blood cells, platelets, and large molecules, they will go into, like salts, amino acids, glucose, vitamins, and nitrogenous waste, and all the other small substances similar to composition of plasma. They will go into the Bowman's capsule as the glomerular filtrate, and this process of filtration is what you call ultrafiltration. Okay, I hope that is clear now. We'll be moving to the next part, selective reabsorption. Yes, the recording, I guess we can send it as a Google Drive link to yours. You can download it. I'll make sure I'll let you know. Then selective reabsorption. This is what I told you that takes place in the PCT and DCT, the process through which useful molecules, ions and water from the glomerular filtrate are recovered and returned to the interstitial fluid and then into the capillary networks. Simply, whatever that was useful but didn't have enough time to be kept back, right? You can't hold back because we are through the pores, all these simple things like glucose, amino acids will get into the glomerular filtrate. What will happen is in the proximal and distal convoluted tubules, these things will be taken back into the interstitial fluid and then into the blood vessels, because those things are not to be removed by as urine. Those things are needed by the body, so they will take them back. Simply, things like glucose, amino acids will be taken back into the blood. That is selective reabsorption. Most of the reabsorption from the glomerular filtrate takes place in the convoluted tubules. Ions, water, and valuable nutrients are reabsorbed either by active or passive transport. What do you mean by active and passive transport, children? Can you tell me the answer? What do you mean by active and passive transport? What's the difference? And yes, interstitial fluid is not blood. Interstitial fluid is also something that comes out of the blood, like whatever, everything except the large substances will come out as plasma, that is interstitial fluid. So whatever that is reabsorbed will come back into the interstitial fluid, and from there it will go into the blood vessels. Exactly. You all know the meaning of active and passive. Very good. Active is using ATP and passive is without energy. For active transport, you need energy. So if there are some substances for which you need energy to take them back into the blood, and some of them you can take them without using any energy. Then the last process of urine formation, that is secretion. The time the seminar will be finished should be around 4:30 to 5, I'm sure, because we have come to almost to the end. We have a little bit more. We have the process of urine formation to discuss and the diseases. After that, if you all have any questions, you all can ask. I will let you to ask questions. And okay, so secretion, we'll go through this as well. If I'm too fast or you need anything for me to explain, please do send me through the chat. I will do it again. Okay, is it clear? Is selective reabsorption clear for you all? Can you all tell me? Okay. Good. Glad if it's clear. Then secretion. The process by which foreign materials and substances not required to the body, including waste, are cleared from the peritubular capillaries and interstitial fluid into the filtrate is called secretion. So I hope you all remember the peritubular capillaries. Those are the capillaries that divide from the efferent arterioles surrounding the tubules. Yeah, so secretion is secreting them out from the blood, simply putting them into the tubules to be removed as urine. That is secretion. Whatever that our body doesn't need, we will secrete it. Okay, first everything was filtered except blood cells, plasma proteins, and platelets. And whatever that was needed to the blood, they took it back through selective reabsorption, and the remaining, which is not needed, they will send it away through secretion. Simply, that is it. Secretion is required because substances may not be entirely filtered due to the short time they remain in the glomerulus. So, just like I told you, there are substances that are not needed to our body. So those substances will be removed. Some substances that couldn't be removed before will be taken away during secretion. Interstitial fluid is the fluid that is found. Now, actually it is something like the internal environment of our bodies. Okay, interstitial fluid is now in our blood. Usually when blood is flowing, sometimes small substances, except blood cells, plasma proteins, and platelets, they will, within the blood, will also go into out of the blood vessels and make a fluid called the interstitial fluid. If you have learned about the lymphatic system during circulatory system, then you would have heard about interstitial fluid, or in circulatory system. Some in our bodies, blood vessels and interstitial fluid are blood and interstitial fluid are separated because we have blood vessels. But in some of the other organisms like arthropods, their blood and interstitial fluid are not separated because they don't have good blood vessels to carry their blood. Okay, so interstitial fluid is something like plasma. And yes, the recordings we can send you to email, or we will inform you that. Then substances that are secreted into the filtrate. Now you have to know the substances that are secreted: H+, ammonia, creatinine, drugs like penicillin or aspirin, and excess K+ are secreted, are removed from the body. Okay, tubular secretion of H+ and ammonia are important to maintain the normal pH in the blood by forming of ammonium. Okay, because blood pH is slightly basic. It's about 7.4, 7.35 to 7.45. The pH is given in respiratory system lesson, I think, when about forming of carbon dioxide, whatever, if the level of pH changed, I think you there was a picture in respiratory system lesson about the pH itself. So to maintain the pH, I told you in the beginning of this lesson, that is why we have this osmoregulation and excretion. So H+ is again acidic, ammonia is basic. So to maintain the pH, ammonium will be formed in the urine and it will be secreted as that. Okay. And secretion occurs in the proximal and convoluted tubules. Secretion may be either active or passive depending on the location or the substance. So depending on where the substance is found. Okay, if it's in the proximal convoluted tubule or the distal convoluted tubule, and the substance, either it's H+ or K+, okay, depending on the substance and where it is produced, secretion will be active or passive. It may need energy or it might not. All right. Okay, then the process of urine formation. Here you have to remember that everything that I've bolded in red is active, everything in blue is passive. That is important because in the textbook, it's a long paragraph. What they have given, it's a long paragraph, and so I have broken them down into slides and points, so it's easy for you all to remember, and I've given them in colors as well. Okay, so process of urine formation. Now we'll discuss this. Listen very well. Okay, this is important because if you ask to write about the process, you have to describe this. And what I'm always telling you, children, is remember everything like a picture. The whole process, try to imagine a picture. Draw a structure of a complete nephron, if you have time, I mean, you should make time. You have to study, right? So take a paper, a blank paper, draw an entire nephron. Okay, what I mean is you have to have a Bowman's capsule and the tubules and the blood vessels. Okay, the peritubular capillaries and vasa recta, everything. And then mark use colors, just like I have use colors and mark whatever. Yeah, secretion also takes place in the tubules. Secretion can take place in the tubules, especially PCT and VCT. And so draw this, just like I was telling you, draw a picture and make sure that you mark everything, what is reabsorbed and what is secreted, right, with the proper colors, mark, and then look at it, study the structure, and keep in so that whenever you are asked to write, you can imagine the structure, you can draw it. If it's an essay, you draw the structure, look at it, and then you can nicely enjoy. Just write it like a story. What we were told when we were doing our exams and we were writing essays was that think as if the examiner doesn't know anything, or think as if you're teaching someone who doesn't know anything, so that you can make them understand. So you have to answer the essay step by step clearly. Okay, like that. Active and passive is not based on the concentration. Active means you're going against the concentration gradient. Passive means you're going along with the concentration gradient. Active and passive mainly depends on where it takes place. Concentration is also, yeah, concentration can, yes, if it's the concentration, yes, concentration can determine if it's active or passive, the way it is secreted or reabsorbed, the concentration of the molecule, and especially the type of the molecule and where it is secreted or reabsorbed determines if it is actively or passively reabsorbed. Okay. So, children, do you all have any connection issues? Or have you all missed any places? Just let me know. All right. Okay, I'm glad you all can hear everything. Okay, so we'll discuss the process of urine formation. I guess some of you all might have had. Okay, so after the process of urine formation, it's like a huge bulk is done. Then we only have to talk about the hormone regulation and the diseases in the kidney, and the lesson will be done. Okay, so process of urine formation. Now I'll explain this. Listen well. Glomerular filtrate, that is in the Bowman's capsule, it has all the substances except blood cells, platelets, and plasma proteins, right? Everything except large molecules will come into the proximal convoluted tubule. Now, from the Bowman's capsule to the proximal convoluted tubule. Then in the proximal convoluted tubule, selective reabsorption of ions, water, and valuable nutrients will take place. So, ions, water, and whatever valuable nutrients will be selectively reabsorbed. Nutrients, especially glucose and amino acids, are actively transported to the interstitial fluid. Okay, especially glucose and amino acids. Now tell me, if you have, or let's say, I will tell it like this: how do you initially identify from the urine of a person if he has diabetes or not? There's a simple test that you can do to identify if a person has diabetes or not by checking his urine. Does any of you know? Exactly, presence of glucose. And get attracted, true enough, because glucose might be present in your blood, and if glucose is present, it means your urine formation process hasn't taken place correctly. So there might be some conditions like diabetes. Okay, so glucose must be 100% completely, glucose and amino acid must be completely removed from the glomerular filtrate back into the blood. It must be completely reabsorbed, okay, actively as well. Glucose and amino acids are actively transported to interstitial fluid, and then sodium plus ions will also actively get transported back into the blood. And because a positive charge in blood, in the cells of the blood, there is a there's a cell membrane, and there is a positive negative balance in the cell membranes of cells. So because a lot of plus ions were taken back into the blood, along with it Cl minus chloride ions, when sodium plus ions were actively taken back into the interstitial fluid, this will drive the passive transport of chloride ions back to blood as well. Okay, so chloride ions will be taken passively, sodium plus ions will be taken back into the blood actively. Okay, is that clear? I hope it's clear. Then the next parts, proximal tubule also will reabsorb K plus ions passively. Now remember, K plus or potassium ion will be passively reabsorbed in the proximal tubule. Okay, remember that, passively. And bicarbonate will also, most of it will be passively reabsorbed. Bicarbonate reabsorption is also important because it also helps in the pH balance, acting as a buffer. Okay, bicarbonate is important in acting as a buffer to maintain the pH of the blood. So bicarbonate will also be taken back into the blood passively. And of course, water will be taken into the blood vessels passively by osmosis. Okay, because we have cell membranes in the cells, osmosis is the method by which water will be absorbed back. All right, so then the last one, a major portion of water reabsorption from the filtrate occurs at the PCT. Remember that a larger portion of reabsorption, selective reabsorption, takes place at the proximal convoluted tubule. Okay. Then now the ones in pink, I have shaded, like I have filled the points in pink for selective reabsorption, and the ones that are in green mean secretion. All right. So okay. So now we're coming to secretion in the tubules. All this time we were talking about selective reabsorption. So things like glucose, amino acids, and sodium were reabsorbed actively. Chlorine was anyway just taken back because of the plus minus problem. Chloride ions were taken back passively into the blood. Then K plus and bicarbonate ions are also taken passively. Bicarbonates are important for buffering of the pH, and water was reabsorbed by osmosis, and a very large portion of selective reabsorption takes place in the PCT. Then the secretion, as the filtrate passes through proximal convoluted tubule, secretion will also take place. Cells lining the cells lining PCT secrete H plus by active transport and ammonia by passive transport. I told you, acting as a for action of a buffer, ammonium will be formed. So H plus will be secreted, removed actively, ammonia passively, and they will form ammonium to trap H plus. Because if there's a large amount of H plus, it will be too much acid, which is not good. Therefore, it will form ammonium acting as a buffer. Buffering means if there is a change in pH, bringing the pH back to normal is buffering. Okay, so for the buffering purpose, ammonium is formed. Then drugs and toxins will also be actively secreted into PCT. Drugs and toxins are not necessary. They have been identified as toxins and drugs by the liver, and then those things will also be removed through the kidneys. As a result of water reabsorption and secretion of different substances, uh, the filtrate becomes more concentrated as it passes through the PCT. So remember, when blood is going through the PCT, because a lot of water is reabsorbed and some substances, solutes are removed, what will happen is the filtrate becomes more concentrated, which means there's less water now in the urine that is about to form. Okay. Now, digestion, it doesn't matter if its digestion is completed first or urine formation takes place. It's, yeah, because now whatever that we have digested in our body, they will be turning into the simpler substances. All the carbohydrates will be turned into monosaccharides, right? Like that, everything will turn into its smaller substances, into the monomers, and then these substances will be taken into the blood vessels, right? So whatever that is taken into the blood vessels, because only the monosaccharides and them can be directly taken into the blood vessels, that is why digestion takes place in your body. I'm answering a question. It says after digestion completed, only whether urine formation takes place. So whatever the end products of digestion will actually result in will be taken to produce urine. Yes. So now I told you in secretion, a lot H plus and ammonia will be secreted, H plus actively, ammonia passively, and so then drugs and toxins also will be actively secreted. The filtrate is very concentrated when it's at the PCT. And now from the PCT, it comes into the descending limb of the loop of Henle. Passive reabsorption of water will continually take place through osmosis, and the filtrate becomes even more concentrated as it comes to the descending limb of the loop of Henle. All right. So then cells lining the PCT secrete H plus by active transport and ammonia. Okay, fine. Next slide. So as I said, when it comes to the descending limb of the loop of Henle, it has become more concentrated. And when we were talking about the loop of Henle, I told you that ascending limb is impermeable to water. All right, ascending limb doesn't let water come to be taken in or out. Therefore, here no water will be reabsorbed. Okay. Okay. Ascending limb is impermeable to water, so no water absorption takes place. But here, a considerable amount of sodium chloride will be taken, will be reabsorbed. Most of sodium is transported through active transport, and because at the ascending limb of loop of Henle, a lot of salt is taken back, but not water, the filtrate will become a little bit diluted. Okay, all this time coming from the proximal convoluted tubule, it was concentrated because water was continuously reabsorbed through osmosis. But once it comes, moves towards the distal convoluted tubule, because the ascending limb of loop of Henle is impermeable to water, what will happen is the filtrate becomes more diluted. All right, up to here, tell me if you understood everything. Or is there anything you missed? You can put on the chat if you understood everything. Did you get everything, children? Okay, I can recap. All right. Okay. Then moving back, this lesson, children, it's not that interesting. I understand, because it has a lot of theory. It has a lot of things to remember, and so it will not be that very interesting to you. But remember that this will be, you will have one MCQ for your MCQ paper, and for structured essay also you will have a part of a structured essay from this lesson, and you might have an essay or a short note. Okay, so it is equally important. And moving on, as the filtrate passes along the collecting duct, now when it comes, no, the distal convoluted tubule. Now from the ascending limb of the loop of Henle, it has come to the distal convoluted tubule. This is playing a very important role in regulating the K plus and NaCl concentration of the body fluids. Okay, so the amount of K plus secreted by active transport. Now, when I was talking about selective reabsorption of K plus, I said that it was passive. I asked you to remember that it was passive, but you can see now when it comes to the distal convoluted tubule, secretion of K plus is active. Selective reabsorption of K plus is passive and secretion of it is active. Okay, so amount of NaCl will be changed according to the need of the body. That is, if the body needs more water, NaCl will not be reabsorbed. But if it doesn't need a lot of water, if the water balance is good in the body, they will absorb or reabsorb sodium chloride in the distal convoluted tubule. And DCT also contributes to pH regulation. Again, in the distal convoluted tubule, H plus and bicarbonate ions will be secreted for acting as a buffer, and passive water reabsorption will be taking place again, unlike in the ascending limb of loop of Henle. And here in the distal convoluted tubule, now hormones will start to act. Okay, I told you there are osmoreceptors in our brain, and for these osmoreceptors, they will get stimulated and they will act on our glands that secrete hormones to for reabsorption or secretion of certain substances. Okay, so aldosterone is secreted by the adrenal gland. Adrenal gland is found above the kidneys. So aldosterone secreted by the adrenal gland stimulates increase of reabsorption of Na plus and water and excretion of K plus. And there's a small mistake here. K plus is secreted or removed. Okay, I have corrected it here in the tute. I think I have given this in blue color. Please make sure to correct it. It must be active. K plus secretion is active. Selective reabsorption is only passive. Here it's correct. In the tute, it might have been given in blue color. If so, then please correct it. So from the distal convoluted tubule, everything goes into the collecting duct. And for bio MCQs, yes, like I told, there is a analysis, as in the first questions up to like 35 or 40. I think they will be asking in order of the chapters. So you will be first asked from the first two chapters, and then it goes on: photosynthesis, respiration, enzymes. You won't be asked MCQs out of the entire syllabus, but it goes in order. You can see if you take a past paper MCQ, it goes goes lesson by lesson. Okay, and then at the end there is a mixed up lesson where you have to select like A, C, D is one or something like that. Until that, everything else is in order. Okay, and we can't say how many questions come from each chapter, but you can just know that there is an order. Okay, so this is the last the last slide of urine formation. Then we can actually do the hormone activity on urine formation and the diseases of kidney, and we can wind up. Now, as this filtrate passes through the collecting duct, it becomes more concentrated. Why? Because a lot of water is reabsorbed. Now, water reabsorption was paused in the ascending limb of loop of Henle. But then again, now after it came to the distal convoluted tubule and the collecting duct, the urine became more and the filtrate became more and more concentrated. And when the filtrate is concentrated, now only urine is formed. Okay, at the collecting duct, urine is formed. Now, at this site, water reabsorption can also be increased under influence of ADH, and urine can become more concentrated. I told you hormones will act in distal convoluted tubule and the collecting duct. ADH, antidiuretic hormone, which is found in the posterior pituitary, secreted by stimulation of the hypothalamus in the brain. You have another part called osmoregulation in homeostasis lesson, which is coming after nervous system. And those who have done it already might know. So the ADH and aldosterone hormones will help in either making the blood more concentrated or urine more or less concentrated. Aldosterone hormone stimulates active reabsorption of sodium and passive reabsorption of water. Because of high urea concentration in the filtrate, some of the urea might even diffuse into the interstitial fluids. If they ask you a question, in which site does urea diffuse into, some of the urea diffuse into the interstitial fluid, that is in the collecting duct. Because there is a high urea concentration in the filtrate, some of the urea will go into the interstitial fluid as well. And remember, urea is not produced in the liver, it's produced in, urea is not produced in the kidney, it's produced in the liver. Final processing of filtrate at the collecting duct forms urine. When producing dilute urine means urine with a lot of water, the kidney actively reabsorbs salts without allowing water to be reabsorbed. So if there is less water if the body wants more water, it will secrete, it won't reabsorb a lot of salts, right? Because when the salt concentration increases, water levels decrease. So if the body wants more water, it will control the amount of reabsorption of salts. Can you tell me if it's clear up to this point? Everything about formation of urine, ultrafiltration, selective reabsorption, and secretion, and the whole process of urine filtration. Is it clear? Okay. All right. Now, the whole big bag part is finished. Describe the role of hormones in regulation of osmotic pressure. Now, we're talking about aldosterone and ADH hormones, okay? Now, the normal blood osmotic pressure is 300 milliosmo per liter. You have to remember the units. If you just write 300, it's wrong. It's 300. Simple M is milli, osmo, OSM is the unit per liter. That is the normal blood osmotic pressure. That is what they have calculated in osmose and given us a value. So I hope you know what positive and negative feedback mechanisms are. You know about feedback mechanisms, negative inhibition, things like that. Have you heard of them? If you have heard, you can put in the chat. You haven't? Okay, so some of you have and some of you haven't. So feedback mechanisms. Here, okay. So here I will describe. Now, when the blood pressure is increased, osmotic refers to salts, the solutes. When the solute concentration is increased, which means the water levels in the body have gone down, this will be detected by the osmoreceptors in the hypothalamus. Hypothalamus is found in your brain. So when the blood flows through the osmoreceptors in your hypothalamus, they will detect it. Don't do that like checkpoints. They will detect it and they will positively act on the hypothalamus. The hypothalamus will go through two pathways. Either it will stimulate your posterior pituitary to secrete ADH, antidiuretic hormone, or otherwise it will generate thirst. Imagine you order something, let's say you order fast food from KFC or Mac or whatever, and then you drink very little water and you sleep. Have you experienced suddenly you will wake up in the middle of the night? It might be you just wake up, or you might see a very bad nightmare, and you will wake up and you will feel very thirsty. Okay, and you will want to drink. You will drink a lot of water, and it will feel really, really good after you have drunk a lot of water. Have you ever felt it, children? Like you wake up and you feel really thirsty. Have you? Can I know? Yeah. Yes, that has happened to me as well. Okay, that is your body saying that you need water. That is your hypothalamus acting on your body to make you feel thirsty. So it will make you wake up with a bad dream, or you make you suddenly wake up, you feel as if you're falling. Some people say it's something called the deja vu, not pretty sure. So actually what happens is the hypothalamus will generate thirst. It will make you thirsty so that you will drink water, and so when your water levels increase, the solute levels will go down and your blood pressure will return to normal. Okay, that is what usually happens. Or otherwise, it will stimulate your posterior pituitary gland. Posterior pituitary is where you find ADH, antidiuretic hormone, that will act on the kidneys, and kidneys will reabsorb a lot of water at the DCT and collecting duct so that you have enough water in your body. Okay, because what has happened is your osmotic pressure has increased as the number of solutes is high. So in order to reduce the number of solutes, you need a lot of water. So these are the mechanisms that your body does to increase the amount of water. Either it will go in a way to wake you up and make you drink a lot of water. Suddenly you will feel very thirsty. Yeah, that is when your hypothalamus makes you thirsty to drink water. Or otherwise, a lot of water will be reabsorbed through your kidneys. All right. So what happens is when your osmotic pressure comes back to normal, until your osmotic pressure comes back to normal, your hypothalamus will keep on positively acting on the pituitary gland to secrete ADH, so that continuously water will be reabsorbed. But that is also not good. Therefore, when your blood comes to normal, blood pressure comes to normal, what will happen is this will negatively inhibit on the hypothalamus, which means this will tell the doesn't tell actually, but there's a process that works with enzymes. The hypothalamus will then stop at stimulating the pituitary so that ADH won't be secreted anymore. Okay, that is negative inhibition. So this is one way using ADH how our body maintains osmotic balance. If it is an essay, no, you can't draw the chart, children. You have to write it in paragraphs. You have to describe the entire thing nicely in an essay. Feeling thirsty under high temperature, yes, because of temperature changes. Maintaining homeostasis, you will learn this in that lesson. Because our body temperature is usually around 37 degrees Celsius, it can't go over that. I mean, it can only be in the range from 36.5 to 37.5. So if you're suddenly put into a heater, let's say, and your body can't tolerate that temperature, then hypothalamus is very important to our body. It only controls a lot of things. It will do these processes to somehow uh, make you maintain that particular temperature and the osmotic pressure in your body. Okay, so this is what is done by ADH. Let's see what aldosterone does. Is this part clear? Whatever is done by ADH, is it clear to you all? Okay, then let's move on to what is done by aldosterone. Okay, here. These two have been given as charts in your resource book. This is exactly what is in the resource book. But if you are asked to write hormonal regulation, please write as in an essay, like with paragraphs. Don't draw charts. All right. I know it's easy to draw a chart, but you're expected to write. Okay, so regulation of osmotic pressure by aldosterone. You can see now you might have normal blood pressure. We'll see now here. Blood pressure or volume drops. Let's say you have dehydration. Okay, you're either in some place where you don't have access to water, or maybe you have diarrhea or something, so you have dehydration, or you faced an accident and you lost a considerable amount of blood. Then what will happen? Sensors in the JGA. JGA is juxtaglomerular apparatus. I have given you a picture in this slide to show the juxtaglomerular apparatus that is found. It's a specialized structure, children, formed by the DCT, distal convoluted tubule, and the afferent arteriole, and it is found closer to the glomerulus. It's formed with association of the distal convoluted tubule and the afferent arteriole of glomerulus. It's apparatus. There are sensors in this juxtaglomerular apparatus, and the function of JGA is that it can secrete certain hormones to constrict our blood vessels. Okay, and that is to somehow get the blood pressure back to normal. Okay, so juxtaglomerular apparatus will detect the decrease in pressure or volume, and they will secrete renin. You know renin. If you have done digestive system, you might have heard of renin. It's an enzyme. And so angiotensinogen is a proteinous hormone that is produced in the liver. Angiotensinogen will be converted to angiotensin 1. You know, if it's given in this way, now in digestive system, also you might have heard of trypsinogen, like that, and they get converted into trypsin later. They're in their inactive forms or dormant forms. You might have heard when you're learning digestive system why they have been given those names. Trypsinogen converts into trypsin later, something like pepsinogen to pepsin, like that. Here also, angiotensinogen converts into angiotensin 1 by the action of renin hormone, renin enzyme. And then there's another hormone called angiotensin converting enzyme that converts angiotensin 1 to angiotensin 2. This is a sort of a long process. Then angiotensin 2 will act on the adrenal gland to secrete aldosterone that will reabsorb more sodium and water increase in the blood pressure and volume, or otherwise angiotensin 2 can constrict the arterioles so that blood pressure will increase, because we want to somehow increase the blood pressure because it has dropped either by dehydration or blood loss. Okay, so all of these processes will take place. It's the JGA, juxtaglomerular apparatus, that detects this loss in pressure or volume, and it will release renin that converts angiotensinogen from liver to angiotensin 1, that gets converted to angiotensin 2 and acts on either the adrenal gland to produce aldosterone or constrict the arterioles, anyway to increase the pressure and the volume of blood. Is that clear? Angiotensinogen is a hormone that is secreted from the liver. Usually, I hope you have not yet learned about hormones. So when you are learning about hormones, they will teach you that hormones are produced in one place and they act in another place. Always it's like that. They are produced in, if you've learned about plant growth regulators, they have, your school or teachers might have taught you the difference between plant growth regulators and normal hormones. Hormones are produced in one place and they go through the blood. They get transported through the blood and they act on another site. So angiotensinogen can be produced from the liver. It can also be produced from the kidney, as far as I know, but it acts in the site of the kidney. Okay, is it clear? Can you let me know, children, if you understood everything up to here? Please let me know. Okay, I'm glad. Okay, so we have done the enzyme activity. Okay, if you need, I'll explain this diagram once again. Now, when the blood pressure drops in the body, either because of dehydration or blood loss, okay, you have to start from the point where blood pressure, if you are asked to write an essay, you have to tell the normal blood pressure of a healthy adult is 300 milliOSM per liter. But due to an accident or heavy loss of blood or dehydration, blood pressure or volume may drop. This drop in pressure or volume is detected by the sensors in the juxtaglomerular apparatus, and it secretes the enzyme renin. Renin converts angiotensinogen hormone from the liver to angiotensin 1, which will be further converted into angiotensin 2 by angiotensin converting enzyme. This angiotensin 2 will either act on the adrenal gland that stimulates the secretion of aldosterone hormone that acts on the distal convoluted tubules and collecting ducts, involving in reabsorption of more sodium plus and water and increase in the blood pressure, or angiotensin 2 by itself will constrict the arterioles to increase the blood pressure, hence bringing the normal blood pressure and volume, that is 300 milliOSM per liter, to normal. Okay, that is how you can write it in an essay. Then other than that, you can explain where is JGA found and all those stuff if you need. But this is the sketch. I just told you how you could write in an essay or a short note if they ask you. All right, so is it clear? Now, did you understand it? Okay. Now we are going to the last few slides. The role of kidney in homeostasis. Kidney is extremely important, okay, because it maintains the electrolytes and water balance in the body. That is, it helps in osmoregulation, and it also helps in excretion of toxic products from our body, whatever that is toxic. Through urine, all these nitrogenous waste products will be removed, and because there are buffers within urine formation, blood pH will be regulated by acid-base balance. Blood volume and pressure will be controlled because of the activity of certain hormones acting on the kidney. And kidney also secretes a hormone called erythropoietin that stimulates red blood cell production. That is very important. They will ask you what is the hormone that is secreted by the kidney. Erythropoietin, that is important in stimulating RBC production. Okay, and also the juxtaglomerular apparatus, I told you it secretes renin, that is important in the control of blood pressure, right? Just like we saw in the diagram. And renin, you might have heard that it involves in the coagulation of milk in the digestive system. So here renin is involved in control of blood pressure. Okay, so kidney is important. Remember at least 3 or 4 of these points, all right? So the importances of kidney in maintaining a constant internal environment. Okay, now disorders related to the human urinary system. There can be bladder and kidney stones, kidney failure, and chronic kidney diseases. Okay, we're going to learn about bladder and kidney stones, kidney failure, chronic kidney diseases, and there's a new disease called chronic kidney disease of unknown etiology. So we're going to learn about them now. Okay, bladder and kidney stones. Now I hope you all are answering the tute. Okay, or at least after the lesson when you get the PDF, please make sure to complete the tute. I'm glad you all are listening till the end. So bladder and kidney stones. Now if you look at the first picture, this man from India, he was some 45 years of age. He had 800 or 890 so amount of stones in his kidney and bladder, right? The doctors have collected all of those, and you can see those are the kidney stones that he had in his bladder. Okay, so we can get kidney and bladder stones because of things like oxalates and phosphates that are in the urine. These kidney stones are also called renal calculi, and when you have kidney stones, it will be very painful. Okay, and that is how you, somewhere around the kidney area, it will feel very painful. That is how you know that you have something wrong with your kidney. I guess he was cured because they took away all of the kidney stones that he had. He might have been in huge pain, right? Having all the stones blocking his whatever ureter, or it must have been painful. So they are also called renal calculi because it goes sometimes things like calcium oxalate. They say it's not good to put tomatoes with fried fish because it can form calcium oxalate, which helps in formation of kidney stones. Okay, moving on. How you can prevent kidney stones is you have to drink a lot of water. All right, because one of the causes is dehydration because not taking enough fluids into the body, and the urine may be alkaline. Okay, and infections, alkaline means basic, so they can form basic substances. Urine is usually acidic, right? If urine is basic or alkaline, then it can be a cause of formation of kidney stones. There can be infections that cause changes in pH of urine. the conditions, metabolic conditions of the body, and maybe family history. If someone in your family had kidney stones, there's a chance for you to have as well. So the only thing that you can do is drink a lot of water, and there are certain foods that you can avoid taking, like I told you, the food that has a lot of calcium, oxalates, and phosphates. If you avoid taking such, or if you limit taking such food, you can actually not get kidney stones. And then kidney failure is the inability of the kidneys to function properly. Therefore, a lot of waste products will be accumulated. The formation of urine will not be taking place properly if someone has kidney failure. Reasons can be diabetes. Now, a lot of people say taking medication for diabetes causes kidney failure. And actually, the reason is it's not the medicine. Usually, the medicine is okay. What happens is diabetes itself can cause, having the condition itself can cause kidney failure, because of it can act on the kidney, and it might be not able to filter the things properly, right? So kidney failure can be fatal. It's a risk factor if you have diabetes, high blood pressure, having family history, and just as you get older, you might have kidney failure. A lot of people, when they're old, you can see that they have kidney failure. And kidney failure can even affect your nervous system sometimes, and it can affect nervous system, those who have kidney failure, and it's harmful for the person. Okay, so then the other one is chronic kidney disease. That is a gradual failing of the kidney. Kidney function is gradually, with time, it is going to ruin your kidney again. Especially diabetes and high blood pressure are the reasons, and having family history, and the fact that you're getting older itself can get you chronic kidney disease. So how do you prevent CKD or chronic kidney disease? Is you shouldn't take a lot of salt. Usually for anyone, a lot of salt isn't good. So it's always better to have a low salt, low fat diet, do proper exercises, have regular checkups, and prevent smoking. Okay. And dialysis. Have you all heard of dialysis? Children, can I know? Have you all heard of dialysis before? Okay. A lot of you have heard about dialysis. So it's a filtration mechanism. You don't have to know a lot about, yeah, exactly, a blood filtration mechanism. You don't have to know a lot about dialysis, just that it's done for patients with kidney failure. There are two types of dialysis. One called hemodialysis and another called peritoneal. If it's hemodialysis, you are connected to a machine. So the machine will purify your blood. If it's peritoneal dialysis, you will have a fluid called dialysate inside your body, okay? And that will draw all the waste out of your blood. Why you do dialysis is because your kidney alone itself can't filter the excretory products with the good products. That is why you need something else to help. Yeah, most of your relations might have done. If someone has kidney failure, they usually have to do dialysis, and before dialysis there will be a minor surgery to do. This will take about 4 hours, the process of dialysis. And the only major risk of dialysis is that you might form a blood clot. Okay, other than that, it's usually what the doctors do when you have kidney failure until you do a kidney transplant surgery. Okay, so they have given you about dialysis as well. Then this one, chronic kidney disease of unknown or uncertain etiology. This is now the major condition in areas like Polonnaruwa in Sri Lanka, especially for the farmers. Okay, because there are a lot of heavy metals found in the fields and water that they drink. Unknown etiology or uncertain etiology is they don't know a proper reason or root cause or the proper symptoms with this disease. Again, here the kidney function is gradually lost over time, and it's associated. People say it's associated with diabetes and high blood pressure. Okay, so the onset of the disease appears to be asymptomatic, which means it doesn't show the symptoms, right? But you could end up with very bad kidneys, especially in places, I've mentioned the places in Sri Lanka where they have this condition, especially in the rural communities with the water that they drink, which has a lot of heavy metals. And as I saw in the news recently, I heard that it's because of the high fluoride levels in water. Okay, in the water that they drink, there's a high fluoride level. CKD and CKDU are sort of different because CKD you will get the symptoms with time, and it will be the gradual loss of kidney, and with the symptoms you can actually know that the person is having a disease like CKD. But CKDU, U is simple. Make sure this U is simple, okay? And for CKDU, that is unknown etiology, means you won't see the symptoms till the end. Until your kidneys are damaged to a stage where it is irreversible, you wouldn't know that you have it. That is CKDU. Mostly found in Sri Lanka. I think in 2019 for the new syllabus, they included this disease condition as well as a new disease that is taking place. If for the news also, they show sometimes that the main reason for this is not fertilizer, but fluoride levels in water that they drink. Okay, moving on. The hypothesized reasons for CKDU. So causes of CKDU can be multifactorial, which means there can be plenty of reasons for CKDU. Either heavy metals like arsenic or cadmium can be the reason, or maybe in rural areas the people, they don't use the hygiene of the people may be quite low, so the food preparing utensils may be of low quality, high fluoride levels, and pesticides that farmers are using in the fields. It might also be because of genetic factors or things like malnutrition or dehydration. The thing is, multifactorial means there can be a lot of reasons. These are hypothesized, which means they haven't found exactly what is the reason for CKDU. So they are whoever the researchers, they are thinking, okay, these might be the reasons for CKDU. So the causes may be based on a lot of factors. And these are the factors that they have pointed out. Okay, there can be heavy metals or metalloids, low quality utensils, high fluoride levels, exposure to pesticides, genetic factors, and malnutrition or dehydration. Okay, now the lesson is done. The entire osmoregulation excretion part is done. I'm glad if you all have written down, or if you can write down this lesson, it would be great, because I sort of included the entire lesson here. So you don't have to go on reading other, questions. I mean, go on reading all the notes that you have, because everything in the resource book is here. I've selected some of the essay type questions that they can ask you from the beginning of the lesson. The relationships between the nitrogenous excretory products and the environment of the organisms. And that is how or which organism secretes what, remember, and where, and the gross structure of the human kidney. So that gross structure of the human kidney, you have to write down the, you have to draw the structure, then you have to mention, like describe the structure. You have to say this is where this kidney is found, the location, and the structure. This is the right kidney slightly lower than the left. Then there is cortex and medulla, and there are you know, the cortex is granulated, medulla is striated. There are pyramids, renal pyramids, and you know, they project into the pelvis to renal papillae. Everything you have to explain, and you can even mention the blood supply. Okay, so that is one complete answer. Then the process of urine formation, I sort of explained how you would write an essay to that, and the role of hormones in regulation of osmotic pressure and fluid retention. That is what we did with the chart. Okay, so can you tell me, was everything clear up to that? Yes, when you are writing about the gross structure, you don't have to mention about the nephrons. The nephron is the deep ultrastructure of the kidney. So if you ask to write about the gross structure, just write down whatever that we have discussed under the gross structure. Okay, it's always okay to write more points, you know, but just make sure that you write everything that is needed. Okay, so any questions, anything you want to clarify, you can ask me now. Okay. So one of you had asked about a paper class. Now, yes, I am about to start classes. So if any of you are interested, I can put my phone number in the chat to everyone. So if any of you want to join my classes, you can drop me a text or give me a call. And if you want, and one of you wanted to know about photosynthesis, if you want to do a seminar, you can talk to online class admins. Yes, I want to talk to you all. This is not yet finished, so wait. And about the stones, it's not like stones on earth. Kidney stones are like stones that are formed out of calcium oxalate and phosphates. Okay, so they're also like stones. They're heavy and they look like stones as well. And for the role of hormones, we have to write both about ADH and aldosterone. Yes, you have to write. If they just generally ask you the role of hormones on maintaining of blood pressure, you have to write about both ADH and aldosterone. And about the recording, we will tell you, I think we can get it as a Google Drive, and we can send it to you. I can either send it through email, or we'll see. We will definitely send you the recording and the PDF as soon as possible. And fluid retention is maintaining or keeping the fluid in. I'm just answering your questions, children. Fluid retention is retaining or keeping the fluid in the body. Okay, so how do your hormones help in keeping it? Something like that. Yeah, you will get the slides as a PDF. Okay, so that you can complete wherever that you missed or where you couldn't write. Okay, so before winding up, yes, and also I want to thank, yes, you wanted a motivation, we will talk about it as well. But before winding up, I want to thank for the beautiful song. It was really nice. The interval, within the interval, I'm glad that we heard it. Thank you very much. And I want to thank online class as well, especially Tarushika, for asking me to do the seminar. And yes, children, structured essays, they can ask you like, what is ultrafiltration, or like explaining the processes of urine formation, and then you want they won't ask you like to write 5 or 6 lines. Okay, maximum number of lines for structure they say that they can give you is about 6 or 7. So they won't ask you long topics to write for structured essays. They will ask you things like what is ultrafiltration, what is selective reabsorption, okay, things like that they might ask you. Okay, and one other thing, children, the thing is, I have made a feedback form, just like the form that you all got to register. There is another feedback form as well. I would love to hear your feedback. If you all have learned anything new, or if it was how it was the presentation clear? Okay, I want to know. So I want all of you all. I'm sure on Tharshika will put it in the group, in your online class group. Please do fill it, children. Just take a few minutes to fill it because I really value your feedback. Okay, I want you all to I want to know things that I can improve. Okay, and please take a few minutes out of your time to do it. So before winding up, I want you all to remember 3 things, children. These 3 quotes have stood up with me. I'm sort of a quote liking person. I like to learn to look at quotes. One thing, there are no shortcuts to any place worth going. Okay, if you want to do something, if you want to achieve something, don't keep looking for feedbacks. It's not only for the exam, it's for your entire life, children. Don't keep searching for easy ways, shortcuts. Just if there is something worth your time and your energy, please don't keep searching for shortcuts. Take the long way, give your maximum and do it. All right, and you, all of you are special. All of you all are capable of more than you know, and don't please anyone else. Whatever you are doing, do it so that you please yourself at the end. Said by Priyanka Chopra, I quote, end of the day, end of the whole thing, you must be able to look back and say, wow, that was one hell of a ride. Make sure that you all work hard and work smart. Don't waste your time, because you have ample time to spend when you have done everything. When you have done, we go back to the first quote. If you have spend your energy and everything on something that is worth achieving, children, then you have ample time and space to enjoy the rest of your life. Okay, or you might feel there are a lot of distractions on your way. You will feel stressed out. I know, I understand, I've been through it, but always make sure that you whatever you want to achieve, if what you're doing right now, if this exam is important to you, make sure that you do it well, smartly, and finish it, and then you can be happy and proud of yourself. You don't need anyone else to say, oh wow, you were superb and stuff. No, you don't need anyone else. It's just the inner happiness you have when you do it well. So just don't let anyone make you feel intimidated or make you feel like you're a failure. No, you're not. It's just you. Okay, if you want something, you get it. That's period. So what I want you to do is start. If you haven't started, start today. Make for biology, short notes are not that successful, but if you can remember things in the making short notes, do make short notes, and always do papers, do past papers. Time, especially timing, is very important. The reason you all get stressed in the exam is because you have not done papers under that particular time period. So when you are doing questions, time them and do. Remember the definitions in a creative way so that you can remember them even under the stressful conditions, because I told you at the beginning, I remember them like questions: why, when, how, like that. And So I hear that you have only 99 days more for the 2021 students. If so, then all the very best. Just don't give up. Do until the end. And if you have anything you want to clarify, okay, you can always ask me. I hope all of you understood the lessons well. So if you want to join my classes, I will put the number in the chat. You can write it down. And okay, I hope it was a good session. And before winding up, I invite Tarashika from online class to end up the session. Thank you, children. Thank you for staying almost 4 hours with me, listening to the lesson. Okay, have a nice day and have a great time, children. Do very well, all the very best. Okay, Tarushika.
Okay, thank you, Miss. Good evening, all of you, my students, dear teacher, and my dear friends. I am here to present the vote of thanks for today's webinar. I would like to thank our guest lecturer, Ms. Parame Gunatunga, for enlightening us with their knowledge. And I think today webinar was full of knowledge and interesting things. And I really appreciate all the hard work and sacrifices you made for this webinar. So thank you very much, Miss. And thank you, my friend, Amana Akandala, for your performance. And my friend, Mr. Dilshan Suravira, who is the group creator and admin of the online class. Thank you very much for your support. And finally, thank you all my friends who joined with us today. Thank you. Have a nice day for all.